Um, yeah, so most of what I'll tell you about is unpublished work. In general, in the past decade or so, my lab has been heavily focused on the whole direction of applying data analysis and machine learning algorithms to medical data. We've been collecting large cohorts of people and tracking them for various purposes and certain indications, including some interventions, which I will tell you about. Um, Really, the first main project that we did, we published already 5 years ago. So I'll talk about it briefly, only because it's also a cohort that we've been working on quite a lot. And this is a project we did in Israel, aimed at trying to understand what healthy nutrition is. Um, and we called it the Personalized Nutrition Project. And in contrast to many other works in nutrition that followed people's weight, after some diet, we decided to focus on blood sugar levels and more specifically changes in blood sugar levels after you eat a meal, because that's directly related to weight management. It has major effect on multiple disease and disease progression like diabetes and other diseases. And what we did was to recruit 1,000 people, connect them to a continuous glucose monitor or CGM, and ask them to log on a mobile app that we developed all their meal intake, so that at the end of the study, we had about 50,000 meals and blood glucose responses to them. And when we have such a response, we can take the 2 hours after a meal, and we can look at the change, the area under the glucose curve, as a single quantitative measure of the response to that meal. And so together with these meal responses, we also collected a lot of data about people. The most novel component at the time, this was 2011 or 2012 when we started, was the microbiome of participants. And this study was published, so I'll just briefly summarize the three key findings. The first is that we showed on this scale of 1,000 people that when you give the same people, different people, sorry, the same food, they have very different responses. So that demonstrates the need for personalizing nutrition. You can see here, anecdotally, 4 participants, when they were given the same bread, and they have reproducible responses, but very different from one another. Um, and this we saw across all foods and all people. And then the next thing that we did was to show that we can actually predict out of sample, of course, using personal information of people, including the microbiome, but not only, various clinical parameters. We could predict quite accurately the responses to meals. So every dot here is a meal, and the x-axis is the area under the curve as measured. The y-axis is the is our predictions, and you can see they match very well. We also, as a third key result in that paper, took a smaller cohort and showed that we can take people who are pre-diabetic, who normally have these huge spikes in glucose levels that you see here across a trace of 1 week, and we could fully balance their blood sugar levels by giving them meals prescribed by the algorithm that are supposed to balance their blood sugar levels. And when we finished this study, one of the key questions was the long-term clinical impact of following such diets, whether they can have clinical benefits. And so we took this challenge upon ourselves, and we did a randomized clinical trial. We recruited 200 people with prediabetes, and we randomized them either to a standard of care diet or a diet based on the algorithm. Because these are people with prediabetes, the primary outcomes of the study were changes in the average glucose levels, measured either by the clinically accepted marker of a measure of hemoglobin A1C, which basically measures your average glucose levels in the past 3 months, and measurements that we did by CGM that we connected to people for the entire half year of intervention. We also looked at the ability to reduce these spikes in glucose levels, so the time spent above this some critical level. And here, anecdotally, is one participant. You can see many spikes in glucose levels prior to the intervention in the 1 month on the habitual diet that probably brought this participant to develop prediabetes. And then on the first month of intervention, we fully balance this person's blood glucose levels. And this is throughout the entire half year of intervention, we see a major improvement. And when we look, not anecdotally, but statistically at the outcomes, these are the outcomes of the standard of care diet. The most interesting panel is here on the left, the changes in the clinically accepted marker of hemoglobin A1C. You can see it goes down after half a year, but in the follow-up, it reverts back to baseline. If we look at what happens on the personalized diet, the algorithm diet, we see double the effect after the half year of intervention. And what's nice is that the effect persists during the next 6 months of follow-up. And the ability to reduce the spikes in glucose levels went from about 30% to about 70% reduction. So for us, this actually concluded about 8 or 9 years of going from idea to doing actually the first, for us as a lab, recruitment of a human cohort in quite a different type of setting of connecting people to glucose monitors, collecting all this personal information about people, developing the algorithm, and then going all the way to a clinical trial that really shows clinical efficacy, and we think can actually be can have therapeutic value for people with prediabetes and people with diabetes, on which we also completed a trial recently. So this was overall a very exciting and very long journey. Another work I want to tell you about that we did on this cohort was actually just published last week, where on the same cohort we measured levels of molecules or metabolites in the blood. And we asked a very fundamental question, which is what determines levels of these metabolites for people? This question is important because many metabolites can have an effect on health and disease. And so we'd like to understand what determines them, be it diet or microbiome, because then if these determinants are modifiable, like diet and microbiome, then we might be able to intervene in them, affect by that the level of the metabolites, and then affect health or disease if these metabolites really are causal to disease. And so on our cohort, we performed measurements of these metabolites. We measured overall 1,200 different metabolites. They are very common ones. 1,100 of them are found in more than half of the cohort. They span a wide range of classes, including lipids, amino acids, xenobiotics, peptides, some unknown compounds. And we thought that as the most stringent way to show that we have a clue on what determines levels of these metabolites, we thought that what we should do is to try and predict them out of sample using the various feature groups that we collected. So as an example, say that we wanted to predict levels of caffeine from dietary data, then we would build a matrix of dietary data, food intake, and we would have the measured level of caffeine, and we would then try to learn this mapping using your favorite machine learning algorithm. We find that actually gradient boosting decision trees are kind of state-of-the-art tools that give really high quality performance. We so far weren't able to find methods that, at least on this size data, perform better. And so then once you have a predictor, you take a subject that whose levels of caffeine you didn't see, but you get this diet data, you make a prediction, you compare it. And in the case of caffeine, these predictions actually work very well. But of course, caffeine is just one of the metabolites that we'd like to predict, and diet is just one of the feature groups from which we would like to predict. And so we basically then built 13,000 predictors for every pair of combination of a metabolite and a feature group, including microbiome, gender, time of day, age, various cardiometabolic parameters, components of the food, and so on. And then the first question that we asked is how many of these metabolites could actually be predicted. And turns out that for over 90% of the metabolites, we could obtain statistically significant out-of-sample predictions. In some cases, the amount of variance that we could predict was high. In other cases, it was low. But for 90% of them, we at least were able to find that one of these components, after you correct for multiple hypotheses, actually generates significant predictions. And if we asked what is driving the predictions the most, then diet predicts by far the largest number of metabolites, microbiome second, and so on. The next question that we asked is how robust are these predictions? And so we collaborated with a group of Tim Spector from the UK, who collected a similarly sized data set of coupled metagenomic and metabolomic samples. And we saw very happily that if we sort our predictions based on the amount of variance that they explain on our data, and we get Tim's data, the microbiome data of UK participants, then despite the microbiome data of people from the UK being quite distinct from the Israeli population on which we trained the model, just as is, we only got Tim's microbiome data. We never got his metabolomic data. We made predictions. We sent them to Tim. He compared it to the actual values of the metabolites that we never saw. And for the top 50 predictions that we made, and knowing which the top predictions they are based on our data, all of them, 100% of them, actually validated on Tim's data. And even if you go to predictions that are that we only explain 5% of their variance on our data, we still get a replication of about 85%. So we're quite confident that these predictions actually are capturing some real associations between, in this case, microbiome and metabolites. Okay, and the next thing we wanted to do was to kind of go under the hood a bit and ask whether beyond just saying, okay, microbiome predicts metabolites or diet predicts metabolites, can we identify the actual specific associations, the drivers of these predictions? And so so Suin is here, and she and her student did really amazing work of bringing forward a feature attribution framework called SHAP. And if you are not familiar with it, I really highly recommend that you use it. The basic idea is that it can take machine learning algorithms, definitely decision trees, and break down the contribution of every feature to the prediction. So it basically tells you how you got from the features to generating a prediction. And it does so in a very nice way that kind of for each feature, it accounts for the contribution of this feature beyond the contribution of all the other features that you also have in the model. And so there's several nice properties to this, but basically that in the end, it's additive. So for every feature, you get its contribution. If you add them all up, you basically get your prediction. So we use this framework to kind of open up our machine learning algorithms and see if we can identify driving features. And so going back to the example of caffeine, if we ask what's the driving feature of this, then as a nice positive control, we got that the single dominant feature is self-reported drinking of coffee is the feature that predicts that drives the prediction of levels of caffeine. So it makes a lot of sense. It's a nice positive control. We similarly can look at two other metabolites. One is a biomarker for citrus fruit consumption. Another is a biomarker for fish consumption. In both cases, the driving features are self-reported consumption of either oranges or fish. So nice positive controls when we talk about diet. And so with this confidence, we can now go to our microbiome-based predictions where we have much less prior knowledge on what we expect. You can see first that here are 4 predictions that are really high, with high predictive power. These are the predictions of metabolites in the blood that we make using only microbiome data. You can see that we make very accurate prediction for these metabolites. Interestingly, the top 2 metabolites are unknown, and now if we apply the SHAP framework, we can identify the driving features as the bacteria that are really driving the predictions. It's different bacteria in each case, and so presumably, if you wanted to modulate levels of these metabolites in the blood because you had reason to think that they may be related to disease or causal for some disease, then presumably by these algorithms and then this SHAP analysis, these would be the metabolites, the microbes, sorry, that you would like to intervene with to either add or subtract in order to modulate levels of these metabolites. So we think that these results really can be a path forward to how one may go about developing microbiome-based therapeutics. So finally, in this work, everything I showed you was by now is so far were model predictions, which are of course correlations, and we wanted to ask whether some of these may actually be causal. So as a proof of concept of that, we took predictions where it's easier to do interventions, again, dietary components. And so we took our model of predicting some metabolites, and we identified those metabolites that, according to our model and the SHAP analysis, revealed that sourdough bread consumption is driving them either positively or negatively. And then we recruited 20 participants, and we gave them for 1 week sourdough bread, and we looked at what happened to these metabolites that were either positively or negatively associated with sourdough bread consumption. And indeed, we saw that after 1 week, metabolites that, according to our predictions, would be positively affected by sourdough bread consumption. Indeed, when you give participants sourdough bread, they increase after 1 week. And this is quite specific because giving the same participants white bread, we don't see this effect. So at least proof of concept that in some cases, these predictions may also be causal. Okay, so I want to move now to other work that we are doing on microbiome and really to a whole new way in which we're analyzing microbiome data, which I think is going to be really the next frontier in microbiome research. And this is basically to go beyond just the composition, which we are all doing now. When you work with microbiome data, you take stool samples, you extract the DNA, you sequence it, you get these short sequencing reads, and then you get a database of bacteria. You can map to that database and you can count and get a picture of the composition of bacteria. Which bacteria do you have? Which bacterial genes or pathways do you have? But of course, when we do that, we have a lot more data because when we do this mapping, we get for every single read, we get whether it actually has any changes, any mutations as compared to the reference. So we can record this variation that we have for each person. And if you think about it, there's maybe thousands of different bacteria. Each bacteria can be millions of base pairs. So in theory, the matrix of data that we're talking about now, it's really big data. So we're talking about the number of people that you have, which let's say it's tens of thousands of people, times, in theory, if you were to sequence very deep and people had a lot of diversity of bacteria, times billions of columns, each with the variant that that person has at any given position. Of course, in practice, this matrix is very sparse because many people do not have all bacteria. And even when you have the bacteria, because of limited sequencing depth, we only sequence to some depth. So of course, this matrix is very sparse, but that's in theory the data that we're analyzing, and we can record, of course, this variation across all bacteria that we look at. And so why is this important? Because we can now, there's several applications of this. We can do metagenome-wide association studies, what we call MWAS, in contrast to genome-wide association studies, where you look for variations in the human genome as they compare to traits. Here we're looking for variation in single base pairs in single specific bacteria and how they associate with various traits in the host. We can also look at transmission of bacteria because we can compare the entire genome of two different samples to see if the genomes are nearly identical, which would mean that it's basically the same. Maybe there was a transmission between these two individuals. And in general, we can study the evolution of the microbiome in the gut across time. So I want to tell you about the application of this MWAS. And so the idea to do this, I would say, is quite straightforward. There's 20 years of work on genome-wide association studies. This is the bread and butter of human genetic studies. And there's a lot of data to do that. There's now companies that have millions of genotyped individuals, and they can do these associations. The problem is that in the microbiome field, we are several years behind, so we only have very small cohorts of hundreds, and cohort of 1,000 people is still considered very large. So this, I think, has been the rate limiting factor. Luckily for us, we've been collaborating with a microbiome-based microbiome consumer company that has even by now over 50,000 metagenomes, about 85% collected in Israel, 15% collected in the US. And this really gives us data that we can begin to do these association studies. So when we do them, if you ever saw a human genetic study, typically people call these Manhattan plots. So typically you would have here in a human genetic study, you would have the chromosomes on the x-axis, and SNPs or positions with variation would be all the different columns, and the row would indicate what is the minus log p-value of the association of variation in that position and the trait that you're looking at. In this case, we're looking at BMI. So in our application, instead of human chromosomes, we have bacteria, and every position represents here a position in a bacteria, and we're associating the variation in that position across all individuals that have that bacteria and have reads from that position. We're associating that with some trait, in this case with BMI. So overall, we were able to analyze 75 million different SNPs in nearly 3,000 species, and over 60,000 of these associations actually turned out as significant, even passing the strict Bonferroni correction. So this is nice, but of course, p-values you can get if you have even small effect sizes, meaning you explain a small fraction of the variation, but you have a lot of data. So what's even more interesting is the size of the effects. So this is a volcano plot showing the same data basically as before, but this time we're putting the x-axis as the size of the effect. So how much of the variation, in this case, how many points of BMI does every SNP explain? And you can see that we have SNPs here that explain several points of BMI. To put this in context, the strongest association of a human SNP is about 1.3 points of BMI. Depending on one's height and gender, I mean, 1 point of BMI can be a lot. It can be even 15 pounds of body weight. And here we're finding several associations that even go beyond the 3, 4, or 5 points of BMI. Zooming in, here's one example. Here's a specific SNP, and you can see 18,000 individuals that have the major allele. They have an average BMI of 27, and you can see here 500 people who have a combination of the major and the minor allele, and they have an average BMI of 31. So 4 points of BMI, this can really be, 40, 60 pounds of body weight difference on average between these two groups. And yet this only single position change in a bacteria is what's explaining this difference from a statistical standpoint. We also saw that these associations, they replicate. We identified them in the Israeli cohort. We showed that they actually replicate. 100% of them go in the same direction of association in the US cohort compared to the direction of association that we saw in the Israeli cohort. So also a nice replication. And then something that's unique about doing meta-genome-wide association studies in bacteria, in contrast to human, is that in bacteria, actually every gene can have multiple different copies because it can have orthologs across different bacteria. So it's as if you're doing the association of the gene again and again and again and again. You're looking at variation in the gene, and then in the related gene in a different bacteria and the related gene in yet another bacteria and so on. And so if you get associations over and over again, you can be more confident that you've really uncovered some true biology. And in this case, I'm showing you a gene that actually breaks down certain sugars, and so this type of association may make sense in terms of having an efficient system for breaking down sugars may provide the host with more more calories from the same exact food, and maybe contribute by that to BMI and obesity. That's speculation, of course, but I think the way that you get to it from the analysis is interesting. Okay, and then here are doing the same thing, but now associations with age. And again, you can see a lot of strong p-values. When we look at the volcano plot, there's actually huge effect sizes. So some SNPs here, and here's a zoom in on one example, can explain a lot of years of age, at least statistically. So you have 16,000 individuals with the major allele. Their average age is, or median age is 54. And you have individuals, 1,400, who have the minor allele, and their average age is 65. Okay, so a difference of about 6 years of age, which is a lot for, again, just a single SNP in a bacteria. Again, this analysis of certain genes, and you can see in this case this DNA gyrase A gene has multiple significant association, and this is an interesting case because this is a gene that's known to mutate in the presence of antibiotics. It confers antibiotic resistance to its host, and this position is actually the position that mutates in a dish when you grow the bacteria in the presence of antibiotics. And so what's nice is that we also got that these positions here, variation in them associates with age. And so we think in this case, basically, this is a marker for either lifetime or recent intake of antibiotics. But it's, I think, very cool that we can identify and derive such a result directly from the analysis. Okay, and the final story, unpublished also story, and another application of this is to look at transmission between strains. So this we did in a context of a disease, in context of atopic dermatitis. And atopic dermatitis, or AD, it's a multifactorial disease. It's very prevalent. It affects about 20% of children, 10% of adults. And because there were clues that it may have a relationship to the microbiome, we decided to do a fecal microbiome transplant on initially a pilot of 9 patients. So every person was his own control. Initially, they received 2 dosings of placebo, followed by dosings of fecal material separated by 2 weeks each. And first, I'll show you the clinical outcome. So this is one participant after the placebo treatment. The clinical manifestations of disease are very obvious. And after the 2 dosings of FMT, you can see really major clinical benefits. Here's another participant, and even weeks after the last FMT, you can still see the clinical improvements. The clinicians we work with tell us that these are not effects that you see in response to known drugs or medications, so I think it's interesting. And if we take their score, the score of the clinicians for the severity of the disease, we can see that after the first 2 dosings of placebo, there's really no change. And after the first even dosing of FMT, we see a major improvement. In fact, overall, on average, a 70% improvement, and 100% of the patients saw some improvement. So very impressive results. And we wanted to see, because this is an FMT, we wanted to see whether we can identify the bacteria that actually are underlying these changes. So We looked initially at baseline and we compared the microbiome of the donor and the patient. At this point, we don't expect to see, the FMT was not done yet, we don't expect to see any correlation. And indeed, when we look at all bacteria, we see that there's a typical distance between the same species across different people. Okay, each person has a slightly different strain, so there's variation. It's between 1% and 0.1% difference across the genome in the copies that any two individuals have of bacteria. And the dots here represent the comparison of these distances and changes between people, between the donors and their recipients. So initially, before the placebo was given, we don't expect and we don't see any relationship between the strain of the donor and the strain of the patient. After we do the placebo, we didn't really give fecal material. We don't expect to see any change, and indeed there is no change. But after we do the actual FMT, we now see that there's a lot of dots here really on the edge at the left. This is our detection limit. This means that essentially we did not find any change in the genome of the recipient and of the host. So we believe that this really represents cases of actual transmission of strains from the donor to the patient. And this is all of this side by side, so you can see. There were also certain bacteria that got transmitted multiple times. So here at the top, there's bacteria here like this Prevotella copri that in all but one of the donors, it was transferred to the recipient. So these may be the common bacteria that confer some of the therapeutic benefit. And then finally, we can take a broader view of the microbiome. So this is by a t-SNE analysis of the distances between samples. And the ellipses here represent samples of the same person, in this case, the donor. And the squares represent the patients, and you can see that they are not so close to each other. The donor samples in this space cluster with each other. And after we do the placebo, there's essentially no real movement between every square here pretty much stays in the same place. But after we do the FMT, we see that now patients become much more similar to their donors. So this is in this case, here's another case, another one, another one, another one, another one, and yet another one. So, so you can see that the changes that we're talking about here after FMT are really huge changes. These are not minor changes. Okay, do we have like 3-4 more minutes?
We have about 5 minutes, Aaron.
5 minutes.
Oh, actually 10 minutes and then 5 minutes for question.
Okay. I won't even need all of that. So these are also, COVID-19 times. And so I wanted to share with you in the last really few minutes just other activities that my lab has been heavily involved with, basically to apply data science and tools to understand numbers in this pandemic and to advise with that advise our government with that on several questions. So we've been involved in several activities. I want to highlight one of them, which is a model that we developed to predict the number of critically ill patients that will have severe patients in hospitals, to understand the burden that we expect to see in the future in hospitals. And the reason we did that was because we had a pretty hard second wave. At some point, Israel led the charts in terms of cases per capita. Our healthcare system defined the threshold of 800 severely ill patients in the ICU simultaneously as a threshold above which the system will start to not respond very well and may reduce the ability to treat patients optimally. So, and eventually you can see here, eventually this capacity was for a week or two was broken, and eventually we had about 900 cases in the hospital. But when this during the second wave, when it became clear that we need to predict the burden, we developed a model. It's actually a very simple model. If you want to predict the number of critically ill patients that you will have simultaneously at the hospital, you basically need to have two components. One component is to learn this conversion from all the positive cases that you've identified. You want to understand how this will unroll in the next week or two in which you're making predictions. How will this unroll over time, across time? How many of the positive cases and during which days they will arrive at the hospital at a critically ill condition. So that's kind of the influx of patients. And you also need to predict the outflux. So how many will be released and recover, and how many will unfortunately die, and how many will move into this ventilation state, because that's a state where you stay longer at the hospital. It can also end with recovery or with death. And so what we did was to just take at any given point historical data and just learn these transition matrices. We separated them by gender and age because they look differently for that. And when you have these two components, the influx and the outflux, you can take the number of patients that you have today. You can add to it the influx of patients. You can subtract from it the kind of simulate what would happen to all the patients that already are at the ICU and those that will be arriving, and by that get some prediction. So using that, there's a few applications. So one of them was we asked, at the time we had about 1,500 cases daily, and we asked, how many cases per day can we even handle before we reach capacity? So when you have a model, you can simulate these things. So we ran through the model different levels of total number of cases a day, and we saw that at our first wave, which was a wave with an older population that got infected, because these are older people, they have the influx is higher. They have a higher chance of becoming critically ill, and so we predicted that at about 2,500 cases we would reach capacity of the system. And on the second wave, which is when we made the predictions, we thought that we could reach above 4,000 cases a day. And at the time, this sounded really crazy because the numbers were only 1,500 and they already sounded very high. And we said, look, even at 4,000, only then you'll start to hit capacity. And actually, this prediction turned out. So unfortunately, it was tested. The number of cases reached even higher numbers. And right when we were around these numbers of maybe somewhat higher than this, on average, at a steady state, we indeed reached the capacity of the system. So I think this model was informative. The other thing that we did was to really play this data science game, and prediction game, but for real. And we all do research in machine learning, and it's quite rare that you really play the game for real, meaning that you make predictions where you really don't have access to the actual result, and you'll have access to it only in the future. So every time that we have all the data, and we always partition it to training and test, it's not the same, and in real life things also change across time. So what we did was to post on Twitter in all of these different dates. We've been doing that for over 3 months now. Post on Twitter, and also give our government these predictions. The predictions are always a range. The gray line here represents the range, and you can see, and the red dot is eventually where the number of critically ill patients was, and you can see that in the vast majority of cases we're actually pretty much on target. And I will say, don't want to go into all the details, but when we missed, like this example here where we thought there would be more critically ill patients, that there were less. When we miss, we actually always learned something from it. So in this example here, for instance, it turns out the healthcare system in Israel probably did experience some form of collapse or reaching capacity. And what happened is that more people who were uh, critically ill, more people stayed at home and didn't reach the hospital, and some people were released earlier. So it's very possible that the actual number during that date was in fact higher. But the fact that you see this miss, it points you to this, and then you start asking questions, and then reports come out on people who stayed home, and so on and so forth. And I think you learn something useful about the disease. So I would say that even failures of the model are sometimes even more informative than when the model works. Um, so this is what I wanted to say about this. And the other thing we did was just about a day before we went into lockdown, we ran the model forward for, we had about 500 critically ill patients at the hospital. We ran the model forward. We saw that that even if you do a lockdown, we know that after lockdown, there's 2.5 weeks that you see a surge in cases. And so we modeled, and then even after that, you'll see the people becoming critically ill. So even if after you do a lockdown, you still have several weeks in which you'll see a rise in cases. We did that simulation, and we saw that at 500 cases, we really are at the last minute, because no matter what we do, if we do a lockdown tomorrow, we'll reach that number. We showed that to the highest officials in Israel, and and they decided, not just based on this, but in combination with other information. So about a day or two later, they declared the second lockdown, and indeed, I think the model turned out to be right, and we reached that number. So with that, I will just put up the list of all of our collaborators from many different places, and of course the amazing group of students that I have, and postdocs and researchers that I have the privilege to work with, and that have been driving all the projects that I showed you today. And with that, I will end, and thank you for your attention.
Okay, thank you. Thank you for the fantastic talk. It was amazing. I see many questions in the Q&A window. So let's see. The first one from Joshua Meyer. Can you comment on efforts to make research from the personalized nutrition project widely applicable? For example, Level Cells, which just raised 12 million for seed funding.
Yes, so I didn't mention this, but the research that we did, when we finished it, our institute licensed it to a startup company. It's called Day 2, and Day 2 is offering this technology as a service, both in Israel and in the US. And this is actually the data set of the microbiome that we analyzed for the microbiome that I showed you before.
Okay, thanks. And then the next question, Queen Yao, two questions. So first, how do you count bacterial SNPs with sequencing errors? And the second one is, do you reference genomes? Do you need a reference genomes, or do you can recover SNPs during genome assembly?
Yeah. So sequencing errors are actually very rare. Illumina sequences with an error of about 0.1%. So typically, just to increase power, we reckon that even a single read, that even if you cover a base pair with a single read, you're better off to call that SNP. And okay, 1 in 1,000 people, it'll be wrong, but in most cases it'll be right, and you're gaining a lot of statistical power. So basically, because we don't really care about any one individual, we care about the associations. I think given this so low sequencing error, we can absorb it. And yes, we do use reference genomes. We're working now on a method to do this in a reference free way, which actually is also yielding interesting and a lot of results that are only partially overlapping with the results that we get from reference genomes.
Okay, great. Okay, so next is from Gil Gerber. What fraction of bugs in the human microbiome do you have reference sequences for? And then are indels an issue or just SNPs?
Yes, so indels and are definitely issues. Like in the human genome, we haven't dealt with them so far. We don't know the fraction of bugs because we don't know what we don't know, but the references that we use are based on assembling very large cohorts of even 10,000 samples. So I think they are pretty representative. We also can map about 80-85% of our sequencing rates. So I think that also tells us, gives us an estimate of how much we're covering. And we are now using all of these samples. We are also making our own assemblies and are going to be mapping to those as well. So I think we'll get a significant expansion of the database we mapped to.
Okay. Okay, so next is from Karen Sachs. Do donor-derived strains persist, or is it repeated administration required? Also, was a course of antibiotics administered prior to transplant?
Yeah, so unlike most FMTs, we did not administer antibiotics. Since we didn't know, we had 4 dosings of FMT. The fact that we saw, I mean, we sequenced several weeks after the last FMT, so that demonstrates that the bugs did persist, at least for this time period. And in some cases, I think I showed you an example of even going 33 weeks forward. So they persisted even at this large time scale. So we don't know yet, but at least with these 4 dosings, the bugs some of them at least seem to persist. And this is a good question. We need to understand this dynamic process better.
Okay, thank you. So next is Jaco. Thank you for the fantastic talk. In the MWAS context, have you also looked at the effect of a gene presence absence within a given species strain or core versus accessory genes?
Yes, so we have, I guess you could say that, well, no, so we didn't really tackle this question yet. We basically, for multiple strains that kind of seem, or species that are similar, we took one representative. So there could be accessory genes that still have a lot of variation that we did not map to. So on the one hand, you can say we have the core, but we also have accessory genes of that specific representative that we took. So I think we're probably looking at some combination.
Awesome. Okay, thank you. All right, so I think I can cover only one question. So could you give a bit more detail on the COVID-19 model you used to learn the transitions?
Yeah, so now we move to a somewhat more complex model of based on Cox regression, but initially it was even just a very, very simple model. So basically just building the table of just estimating frequencies of transitions from a PCR positive test to a status of critically ill in the hospital, just frequencies of age and gender specific frequencies across days following a PCR positive test. So just this frequency table is basically an initial simple model that we use.
Okay, great, thank you. I forgot to mention the last question was from Safia Seli. Okay, so we have a couple more questions, but I think we really need to turn to the next oral session. It's already 2 minutes past. Okay, let's thank the speaker. It was amazing talk and then really good answers to the questions. And thank you again.
Thank you. Thanks a lot.
Okay, so okay, so So now we turn to the next speaker. So it's the first oral session today. So Asia Waria from University of Washington is going to talk about her machine learning approach to select binding peptide aptamer pairs. Asia Waria.
Great. Let me share my screen. Um, 1 second.
Mm. And I don't think you shared the screen. Okay.
Oh yeah, there we go.
Yeah.
Um, all right, can everybody see this?
Yeah, it looks great.
Great. Hi everyone, my name is Aishwarya. I'm a first-year graduate student at Princeton University, but today I'll be presenting work done at the University of Washington. Our work is titled Molecular Matchmaker, Selecting Binding Peptide Aptamer Pairs Using Machine Learning. And this work was done in the Missile Lab at the University of Washington. So predicting molecular interactions can be really useful. For example, one way to recognize the presence of a particular target peptide is to use an aptamer designed to bind to that peptide with high specificity. As you can see, this can have an impact in drug design, diagnostics, immunotherapy, and in numerous other fields. Current methods to do this work, including CellX, which you can see a figure of on the right, are fairly time consuming. They rely on wet lab experimentation, and they're often not generalizable. Improving the CellX pipeline can have a dramatic effect on selecting these peptide binding aptamers. So before we go any further, it's important to define what an aptamer and a peptide is. So for the purposes of this work, our aptamers are 40 DNA nucleotide long sequences, and they are generated uniformly. So that means that at every position, it is equally likely to be a T, G, or C. Our peptides are short peptides that are 8 amino acids long, and they are generating using something called the NNK distribution. NNK is kind of out of the scope of this talk, but standard distribution of peptides. And it's like a probability distribution that you can see on the right over here. So for this work, we have 2 research questions. One is, how likely is it that a given peptide and a given aptamer will bind? And the second is, given a target peptide, can we find an aptamer that will bind to it? So there are a couple of groups that have worked on similar problems with predicting binding affinity between molecules, and a few aspects of their methodology led us to structure our experiments a little bit differently. So the first thing is that their training sets are often pretty small, and you can see that that would lead to a model overfitting. The second is that they train using positive data, so positive biologically validated binding data, and then they computationally or randomly generate negative data. And what this leads to, like, as an unfortunate side effect, is that the models that predict binding affinity actually learn to distinguish between the data generation characteristics as well as the positive, like the characteristics of the positive data. So if you approach it in that way, you're not actually learning the molecular interactions of binding. You're actually learning, the way that you generated negative data. The last is that many of these approaches define binding affinity as a binary metric. So 0 if it doesn't bind and 1 if it does bind. Now, binding affinity, when it's measured empirically, is measured in something called nanomolars. And you can see that that is like on a scale. And so when you treat binding affinity as a binary measure, you actually gloss over a lot of the importance of like that scale between certain molecules. So we contribute 2 things. One is a set of data sets. So we have 2 data sets, one positive and one negative. And both are the result of wet lab experimentation. The second is that we introduce classification benchmarks, and these are preliminary neural networks to predict binding affinity. So now into the first contribution. So we have two biological data sets. One is binding and one is non-binding. Each data set has 4 IID experimental replicates. So you can think about them as 4 independent data sets that are binding and 4 independent data sets that are non-binding. Our binding examples are pairs that may have interacted, so one aptamer and one peptide that may have interacted. And our non-binding examples are pairs that may not have interacted. So again, same thing, one aptamer and one peptide that may not have interacted. Now you can see that there's a little bit of uncertainty in this interaction. And for this reason, I think it's important to go over the way that we generated this data set. So our experimental process is called selection of non-binary. We mix a large pool of DNA aptamers, and we mix that with something called mRNA labeled peptides. And we put this through high throughput sequencing, and then we take what is called a molecular snapshot. This molecular snapshot basically associates aptamers and peptides that are close together. And what we hypothesize is that aptamer peptide pairs that are close together because they have a higher affinity for binding with each other. Now you can see that there is a non-zero possibility that an aptamer peptide pair will be close to each other by chance, and we call this random diffusion, or for other reasons that are not really specific to aptamer peptide binding. So the result of this experiment is a set of aptamer peptide pairs that may or may not have interacted due to high affinity aptamer peptide interactions. Now the negative experiment, so non-binding aptamers differs in one way, and that's that we only use mRNA sequences for mixing with the aptamer library instead of mRNA labeled peptides. And we put this through high throughput sequencing again. And what we're associating at the end is aptamers with mRNA sequences that we can later translate or in further peptide translation. We treat this as our negative data set. The other thing that we use is one computationally generated data set. So the space of aptamer peptide interactions, 40 length aptamers and 8 length amino acid sequences, peptides, is pretty large. And so we know that a large like subset of these interactions is highly unlikely to bind. So based on that, a randomly generated pair is also highly unlikely to bind. And so we computationally generate this data set using the background distributions of our biological data sets, so NNK and uniform. And we use this as a supplement to the positive and negative data sets that we biologically validated while we're training. The second thing that we introduce is our classification benchmarks. So when I talk about our classification benchmarks, I'll go into how we derive our objective function, the way that we encode our molecules, our network architectures, and some results. So before we talk about the actual objective function, it's important to understand the constraints that went into designing this. So as I mentioned earlier, our data set is not a perfect set of binding and non-binding pairs. There's some non-zero probability that a binding pair or a pair that's in our binding data set is actually non-binding and vice versa. The second thing, which I mentioned briefly, is that the search space for all peptide aptamer interactions is extremely large. So if there are 4 nucleotides at each position, that means there are 4 to the 40 possible aptamers. And if there's 20 potential amino acids at each position, that means that there's 20 to the 8 potential peptide sequences. And so this interaction space is extremely large. What we do know is that binding is a very rare event in this search space. So ideally, a model that says that that a given aptamer peptide pair is binding does so on a pretty rare basis. So given those constraints, let's derive this objective function. So let X be a peptide and let Y be an aptamer. What we want as a model, F sub theta, is a model that takes in X and Y and outputs the probability that X and Y will bind. So notice that we're not treating binding affinity as a binary measure, and instead we're looking at a probability, so a value between 0 and 1. You can think of F sub theta as a function parameterized by theta. And so this could be like a neural network with the weights theta. So what we're going to do is we're going to do a weighted cross entropy loss, and we're going to add extra penalties to discourage certain behavior. So you can see on the second line over here that we have a hyperparameter called alpha. And what this does is it discourages our model from classifying examples in our negative data set as positive. And then we have another hyperparameter called beta over here, which discourages our model from classifying a random sample as positive. We can solve this objective function using stochastic gradient descent. But first, we need a way to convert our aptamers and peptides into numerical representations. So our aptamers for all of our experiments are 1-hot encoded, and if there are 4 possible nucleotides at each position, you can see how this would generate a 40×4 matrix. Our peptides, we experiment with 2 encoding strategies. One is 1-hot encoded, so this results in an 8×20 matrix. And the second is something that we call BLOSUM 2 encoded. So the BLOSUM 62 matrix is a similarity matrix between that quantifies the similarity between a given amino acid and every other amino acid. And so you can see how the BLOSUM 62 matrix would be a 20×20 representation. The way that we encode an amino acid is by taking its respective row in the BLOSUM 62 matrix. So that would also result in an 8×20 matrix. So we decided to use as our benchmark architectures very simple architecture. So we have a linear baseline, which only has 1 layer. We have a convolutional baseline, which has 1 convolutional layer. And then we present two-headed architectures. And you can see what that would look like on the right. This is just a rough characterization. Our network architectures, we want them to be as simple as possible to quantify how easy it is to make these distinctions between positive and negative pairs. For our training, we have 3 hyperparameters to learn. We have a learning rate, an alpha, and a beta. And like I mentioned earlier, we'll use stochastic gradient descent. When we train, we will train with positive biological data, negative biological data, and that computationally generated data set. When we test, we're making 2 comparisons. One is between positive and negative data, and then one is between positive and generated data. So we treat that generated data as negative, but we want to see our model's abilities to make these 2 distinctions. We train using replicates 1 and 2, so that's 1 and 2 in the positive data set and 1 and 2 in the negative data set. And then we test using replicates 3 and 4. So I'll present results on 3 and 4. So here are the results on replicate 3 and 4. So the first figure that you see on the left is the distinction. Our models are trying to make the distinction between positive and generated data. And you can see our models actually do fairly well on this task, given that they're pretty simple and that like every single one of them does better than average, which is our dashed black line in the middle. Now it's much more difficult to distinguish. Our models have a much more difficult time to distinguish between positive and negative data. So you can see that they all have AUCs that are greater than 0.5, but they're still pretty close to 0.5. So our simple models have a difficult time with this. You can see similar results on replicate 4. We do slightly better on the positive generated data than in replicate 3, and then we see the same type of like the same phenomenon where our simple models have a difficult time distinguishing between positive and negative data. So based on our results, we think that there's promising future work in 3 directions. First is exploring molecule encoding strategies as well as new network architectures. These are pretty simple baselines, and we think that there's a lot of work to be done in understanding what architectures help distinguish between that positive and negative category. The second is once the models are in a good state, we can do additional aptamer generation experiments. So we've actually started this work already. What we're trying to do is understand how to optimize over the aptamer search space. So let's fix a target peptide and let's fix a model, like a surrogate model that predicts binding affinity. How can we optimize over the aptamer space to find an aptamer that predicts or that pairs with that target peptide and results in the highest probability of binding? The third thing is to biologically validate binding and non-binding predictions. And so we're fortunate in the missile lab to wet lab where we can validate the predictions that our models make. And this kind of corresponds to both just predicting binding affinity, but then also with the aptamer generation experiments. So if our algorithm suggests an aptamer, we can validate that. So in conclusion, we want to improve the CELX pipeline. And in order to do so, we introduce data sets that characterize peptide aptamer binding and non-binding interactions. And we introduce classification benchmarks to enable future work in this area. I'd like to acknowledge my collaborators, Yu-Hao Wan, Jeff Navala, Kevin Jameson, Louis Seze, Karen Zhang, and Phil Young. And a special thank you to the Missile Lab at the University of Washington and the Paul G. Allen School of Computer Science and Engineering. I'm happy to take questions now.
Okay, thank you, Aishwarya. So let's see. Is there any questions from the audience? Yeah? Okay. All right. Oh, so someone asked the question, but not in the Q&A. Let me just read it. It's from, it's hard to read the name here. So someone posted a question in chat. So have you tried learning the encodings for the residues?
Yeah, we haven't really tried learning the encodings. I think we left that as future work. What we were going for is, can we learn as simple of a representation? So there's a lot of work, additional work in that area.
Okay, so so one question from Safia Selig. Is the generated data balanced between positive and negative?
So what we do with the generated data is we kind of treat it as negative. The reason is because like it's highly unlikely for a randomly generated peptide aptamer pair to actually bind. And so we just assume that it doesn't bind.
Okay. All right. Thank you so much, Aishwarya. So we will turn to the next speaker.
Do you want me to do this, Suin? I think it's my turn to take over.
My time to
Hi, everyone. I'm David Knowles. I'm one of the co-organizers. So next up, we have Surag Nair, who will be talking about fast ISM, performant in silico saturation mutagenesis for convolutional neural networks. Take it away.
Thanks, David. Everything okay?
Yes, looks good.
Sweet. Thanks. Hi, everyone. Thanks for joining. I'm Surag from Anshul Kundaji's lab at Stanford. And this is some fun work I did with Avanti in the lab. Avanti had a great talk yesterday, so if you missed that, make sure to check out the recording. But this is about us speeding up in silico saturation mutagenesis for convolutional neural networks. And we did this by leveraging a few simple insights that I hope by the end of this talk would be clear and kind of obvious even to most of you. So a common task in computational biology is to take raw sequence, for example, DNA or even protein sequence, and to map it to an associated functional readout, typically something that comes from a high throughput sequencing assay, such as a TAC-seq, ChIP-seq, DNA-seq. And the task usually would involve predicting a binary presence or absence of features like peaks, a quantitative prediction like the number of reads in peaks, or in the case of more recent models like the BPNet architecture, predicting the profile of the actual peak itself. All of these models start with a one-hot encoding of the sequence. And as the name suggests, convolutional neural networks start with a convolution filter, which is nothing but a simple matrix that scans across the input sequence. At each position as it's scanning along, it computes a dot product between the sequence of that position and the filter itself and outputs one scalar value. Now, these filters are essentially like position weight matrices. And in fact, after learning is complete in these models, a lot of them seem to capture either partially or completely a bunch of known motifs. A convolution layer consists of multiple such filters. And so we get multiple channels in the output of our first convolution. Typically, convolution layers are followed by max pooling layers, which essentially split the input in blocks of a fixed size, in this case, a size of 3, and compute the max values across each channel. And that's why you see darker values here. Typically, the output of a max pooling layer goes through a nonlinear activation function like ReLU, batch normalization layers. I've omitted those here for simplicity, but you can then repeat this convolution and a max pooling block multiple times, and in the end, flatten the output of the final convolution layer, pass that through one or many fully connected layers, and then get the final desired output. And like any other machine learning model, you can train it with an appropriate loss function that encourages the model to learn the signal that's desired. Now, a common downstream task with once you've trained these models is to understand how mutations or variants in the input sequence affect the output. And a simple way to do this is known as in silico saturation mutagenesis. And the way it works is pretty straightforward. So what happens is, if you look at the left side here, we start with the first mutation, which is an A to C. And then the first convolution filter scans across the input sequence, followed by the subsequent layers. And the final output of the model is then captured. We then try the A to G mutation, again, scan the convolution filter, compute subsequent layers, and then record the output as well. And so this goes on. And so we try the A to T mutation, after which we move on to the second position, try the C to A, then the C to G, C to T, and so on and so forth. And we scan across the entire sequence right till the end, inducing every possible mutation and recording the output. Now, as you can see, this is extremely time consuming because for a single input sequence, you're computing multiple forward propagations. In this case, you're computing forward propagations 3 times the length of the input sequence, which is insane because when you're even training a model for say 50 epochs, you would do 50 forward and backward propagations through it. But here you're doing 3,000, if you have a 1,000 length input sequence model, you're doing about 3,000 forward propagations. But nonetheless, ISM gives us positional contributions to the output and effects of variance, and also doubles up as a feature attribution method. So in this figure here that I borrowed from David Kelly's paper, a model that predicts the DNA signal in H1HESE cell type shows that there's a certain subsequence that has a height and importance score that in this case codes for the FOSS motif. So I'm sure some of you might have noticed in the last few slides that when we do induce a mutation, for example, a G to T mutation in the third position, and scan the first convolution filter, a lot of the computations that are involved actually do not change from the reference sequence. So in this case, as you scan the first convolution filter, only the first 3 outputs change, and none of the other outputs change. And that's because the sequence really hasn't changed at any position except the positions influenced by the mutation that we've changed. Similarly, if you look at the 5th position from the right, none of the values change when compared to the reference sequence except the last 5 values. And these values correspond to the positions where the filter overlaps with the mutation that we've induced. And similarly, if you look at a mutation induced in the middle of the sequence, as the first convolution filter slides along, only 9 positions in the output are changed. And these 9 positions correspond to the width of the convolution kernel in this case. So essentially what you can see is at the end of the first convolution, only a small fraction of the entire sequence is actually perturbed when compared to the reference sequence. And to appreciate this better, think about the case of that we usually encounter in typical models that take in sequences of a length 1,000 or even 2,000 base pairs as input. In models like that, the output of the first convolution, only a very small fraction of the entire intermediate output is actually perturbed at the end of the first convolution. And you can track the regions that are perturbed across all of these intermediate outputs right till when you flatten the output. And in fact, in a lot of these cases, what happens is that even after you flatten the output to a vector, only a fraction of the neurons in the flattened output are actually perturbed compared to the original vector you'd see on an unperturbed original sequence. Once you run the fully connected layers, however, you see that all of the neurons are perturbed, and that's because each neuron depends on all the neurons in the input, And so long as there's even a single input, single neuron in the input that is perturbed, the output neuron will also be perturbed. And so what FastISM does really is leverages this observation and caches all of these intermediate outputs. So what we do is on the unperturbed input sequence, we do a forward propagation and cache all of these intermediate outputs. And then when we encounter a specific mutation, we localize our attention only to paths that need to be recomputed, and then use the cached values shown here in light blue and light green as required, and kind of stitch them together with the actual perturbed values that are computed. And in doing so, we end up saving a lot of time in the convolution layers. Um, Although in the final fully connected layers, we need to compute pretty much the same number of values that we did before in a standard implementation of ISM. The reason this works really well is that in a typical convolutional neural network such as this one, over 90 to 95% of the time is actually spent in the convolution layers and not in the fully connected layers. And so if you if you save a lot of time in the convolution layers, that consequently leads to a lot of savings in ISM. And so we've implemented FastISM as a KNUS package, works with TensorFlow 2.3 and above, and it's got a very painless API. And what you can do is you can just import and then wrap your model, your sequence model with this function. And then as you iterate over your sequences, you can call the FastISM model just like you would call any other model. We benchmark FastISM for a few common architectures like the Bassett architecture, factorized Bassett architecture, which is a modification of the Bassett architecture, and the BPNet architecture, which predicts profiles. And to do so, we looked at the time taken to train the model for 50 epochs, to perform standard ISM, followed by performing FastISM for every 100 megabases of sequence content. So what you can see is when you train these models, they take on average about the same time, maybe a factor of 1 or 2 within each other, but they take about 1 to 2 hours per 100 megabases of sequence content. Now, if you were to interpret the same number of sequences, the same amount of sequences with a standard ISM input, it's crazy because it takes at least 10 to 20x more time for the same number of sequences. And even if you were to interpret, say, a fraction, a fifth or a tenth of these sequences, you'd still be spending more time to perform ISM than training the models, which by itself is usually a slow step. With fast ISM, however, we're able to significantly reduce a lot of these times. And in fact, for models like the Bassett and factorized Bassett architectures, which have input sequence lengths of 1,000 and 2,000, we get over 10x improvements in speed. And this brings it much closer to train time for these models. For the BPNet models, the improvements are a bit more modest, and we see about 2x improvements for a model of length 2,000. But the big picture is that these speed ups do depend on the exact model specifications. For a lot of commonly used models, you see significant gains. We've also tried benchmarking on DeepC and Basenji models, and we see significant 5x and above gains. Another class of feature attribution methods are these backpropagation-based method approaches. And the way they work is by first selecting a specific output in the final layer, and then backpropagating to get the derivative of this output with respect to each of the inputs. So since this is a very commonly used feature attribution technique, we also benchmarked FastISM against a few common methods, such as integrated gradients and DeepShap. And what we see is that for models that have a single output, for example, the Bassett and factorized Bassett architectures, in which, in this case, we force them to have a single output, we see that FastISM is in the ballpark of these other feature-based, backprop-based feature attribution methods. And if it's slow, maybe about 2x, or in this case, 4x. However, if you look at models like BPNet, which have multiple outputs, for these backprop-based methods, the method needs to be called in a loop for each of the outputs. And so as a result, these methods are absolutely infeasible. And so BPNet really shows that when you're looking at multiple outputs, FastISM is the method of choice for interpreting those sequences. So I want to end with a discussion on ISM and backprop-based methods. Both have their pros and cons and have slightly different use cases. ISM specifically is more important when you're scoring mutations, because the absolute values that ISM gives you are meaningful, as they're the predicted change in the outputs. For backpropagation methods, the absolute values by themselves are not necessarily interpretable, although the relative values are. ISM does not explicitly require any reference sequence apart from the sequence itself, whereas backprop-based methods use dinucleotide shuffled sequences as reference, which can be a little ad hoc, as the exact influence of the kind of references one uses has not really been interrogated and investigated. When used as a feature attribution method, ISM does have a drawback in that it suffers from saturation effects, unlike some of the other recent methods like deep lift and integrated gradients. ISM implicitly handles multi-output models, as I'd shown you in the previous slide, whereas backprop-based methods need to be run for each input in a loop. Computation time for a standard implementation of ISM is fairly slow compared to backprop-based methods, but with fast ISM, these are now comparable, and so computation time is not really a deterrent to use ISM-like methods. So in conclusion, if you wish to do an ISM-like analysis, I'd love it if you give fast ISM a try. Within 5 lines of code or less, you can get started. It's pretty painless to use. It's easily available to download on PyPy using pip install. The code's on GitHub and we have a preprint out. And yeah, if you find any bugs, please let me know and I'd be happy to help you out. With that, I'm happy to take questions.
Yeah, great. Thank you so much. And so reminder to put your questions in the Q&A. There's 2 questions right now. So Li Shen says, does fast ISM work only with Keras?
Yeah, so as of now, it works only with Keras, but there's nothing as such stopping it from being re-implemented in another language, another framework, say PyTorch. It was just easier for now to implement it in Keras. So.
Great. Safia Silik says, how can you extend this technique to predict for indels? That's a tough one.
So there might be a few issues when we're looking at, so I think it's possible. Yeah, I think I haven't looked at it, but at least for small insertions of say a few bases, I think the same benefits should apply. One caveat though is that fast ISM does not work for single SNPs. It works when you're doing a full saturated mutagenesis, because it necessarily computes one full forward propagation on the input sequence and then multiple smaller propagations on parts of the mutated sequences. So if you want to do an insertion at say 10 or 100 positions in the middle of the sequence, then it could work. But if you want to do it for a few selected sequences at a few selected positions, then you might not see any gains.
Right. And Jacob Bartos Ziebrich says, do you support inputs/alphabets other than DNA?
Yeah, absolutely. I think it's a pretty general implementation. You can use RNA or proteins. It doesn't even have to be a single mutation. You can mutate a range of inputs, say 5 bases or 5 positions of input. And there's a bunch of things that's pretty well documented. You can give it a shot.
I had a couple of questions, and other people can see if they have any more while I'm asking these. So is there an intuition for why the gain relative to BPNet is so much less than the relative to Basenji and Bassett?
Yeah, the key idea there is that it uses a lot of dilated convolutions. Right. And so with exponentially increasing dilation rates, so what happens is after 4 or 5 layers, it's essentially the range mutated by a single sequence spans, say, half of the intermediate output of that layer or even more. It is possible to get slightly better improvements than what I've shown you. I haven't really optimized it for that use case. So, but that's the main idea, and it's limited. So there's an upper bound based on the cone or the fraction of positions that are mutated are affected rather.
So I don't know how much sense this would make for genomics applications, but in computer vision, there's all this work on robust adversarial training, right? So putting in effectively what to us would be mutations, like can you change one pixel and then change the prediction that a model would make, or ideally you want models that are robust to those sorts of intuitively small changes. So if you thought about whether you could use this within a robust adversarial training approach?
I have not actually. I think that's a pretty cool idea because the same idea is amenable to a two-dimensional version in which you start with images. So no, but that's a great suggestion.
Yeah, I think that was everything I had, and we're just ahead of schedule. So thank you for staying on time. Thanks for a great talk. So next up we have, I think, I have a number of authors listed here, but I think Mohammed Sabarayan, are you presenting?
Yeah, that would be me.
For this, great. Just want to check that your screen sharing will work. It's a long title, but drug repurposing for COVID. So I will let you give the whole title. Thank you.
Thank you. So can you see my pointer as well?
Yes, we can.
Okay, great. So, okay. Hello, everyone. My name is Taber, and today on behalf of my colleagues from SFU and UBC in Vancouver, I'm going to present our latest work on drug repurposing efficacy estimation based on morphological analysis of SARS-CoV-2 infected cells within a multiple instance learning framework. The COVID-19 global pandemic has urged the research community to focus on SARS-CoV-2 infection for therapeutics and treatments. There has been multiple studies for drug repurposing, and there has been some great news within the past weeks about the vaccines. The drug repurposing aims to identify existing clinically approved treatments that could be redirected toward new diseases. High throughput morphological analysis of cells stained with multiple fluorescent dyes offers a rapid approach for antiviral screening. Morphological analysis uses the morphological features from single cell or cell populations as an observable variable to draw conclusions about the cellular dynamics underlying. Morphological features can be extracted from images of infected and uninfected cells, and then applied to the images of infected treated cells to predict antiviral efficacy based on the cellular morphology. We have two goals in this work. Our first goal is to localize the infected cells within a sample cell population without using any specific molecular tools for tracking the virus by training a model that distinguishes between infected and uninfected sample images. These localized cells can later be used for inspection to better understand the morphology and the biology of the infection. Second, we seek to estimate the efficacy of treatments based on the morphological features that so that we can assess a wide library of FDA-approved treatments for a high throughput screening. We hypothesize that an effective treatment causes drug-treated infected cells to look morphologically similar to uninfected cells. So if we somehow manage to extract morphological features induced by the SARS infection to the cells, then we can use them to come up with a measure that allows us to compare a treated infected cell to a healthy one and quantify how effective a treatment is against SARS-CoV-2. We propose a method that consists of two components. A component that is able to classify images into uninfected and infected class to learn the morphological features that are induced by the SARS-CoV-2 infection to the cells, and a component to estimate the compound efficacy against SARS-CoV-2 based on the morphological features that we extracted and learned in the first component using a statistical model. I will dive deeper into the details of these two in later slides. But in order to be able to design a model that is able to extract the morphological features that we are looking for, we need to understand how an infectious agent spreads in a sample cell population. When a sample cell population is infected with a virus, only a proportion of the cells are infected and not all of them. In the microscopy images of these samples, there are no tags or probes for the virus, especially if these images were created at very earliest stages of an outbreak, such as what we had with the COVID-19. So there are no pixel level annotations that indicate which cells are infected that we can use to extract and learn the morphological features that we are looking for. The only label that we have is whether the sample population is infected or not. So we ask, is there a way to identify infected cells just by using these labels? We found that the answer is yes, and it can be done using multiple instance learning. Multiple instance learning, or MIL, is proposed as a variation of supervised learning for problems with incomplete knowledge about the labels of the samples. In typical supervised learning, every training sample or instance is assigned with a label. On the other hand, in the MIL, we only have the labels assigned to a set of multiple instances that we call them bags, and there are no labels for individual instances. The goal of MIL is to classify unseen bags or instances based on the bags that it had seen during its training. So a simple example will help us better understand this framework. Consider that there is a secret room and we have multiple sets of keys. And for each of these sets, we know that whether we can open the secret door or not. The MIL tries to answer two questions here. Given a new set of keys, can I enter the secret room? And what is the magic key or magic keys that open this secret door? There are multiple assumptions about the relation between the instances and the labels. However, the standard assumption is that in a binary case, a bag is labeled positive if at least one instance in the bag is positive, and the bag is labeled negative if all of the instances in that bag is negative. With that in mind, we can use this framework to localize SARS-CoV-2 infected cells within a sample population in a fluorescent microscopy image. So if we break down the sample image into multiple smaller patches, we can think of these patches as instances and the sample images as bags. Similar to the secret room example, we have sample images that are infected and we have the ones that are not infected. And we are trying to find the answers to two questions. Is this sample cell population infected or uninfected? And if so, where are the infected cells? Based on the standard assumption that we have, we assume that a sample population is infected, then there should be at least one patch that contains an infected cell, and an uninfected sample cell population doesn't have any infected cells. With that being said, we can use a deep neural network as a learner in an MIL context. For each sample image, we break it into smaller overlapping patches. Then these patches are fed into the neural net. Here we use the resident 50 for a binary classification between uninfected and infected. The output of the model is the probability of a patch being infected. Then for each sample image, all of the patches are sorted based on the infection probability, and the k-top patches are selected for training. These patches are assigned with the image sample label that we have. A cross-entropy-based loss is used for the classification, and the weights are updated accordingly. So within each iteration of the training, we filter the instances based on their infection probability, and we only use the ones that we have confidence in their label to train our instance classifier. We use the RxRx19 dataset provided by the Recursion Pharmaceuticals in this work. It includes monolayers of normal human renal cortical epithelial cells in 3 viral conditions. MOC, which is the healthy control samples, UV inactivated, which are the samples that were infected with the UV irradiated inactivated virus. And the third group is active SARS-CoV-2. The active SARS-CoV-2 is further divided into 2 subgroups, untreated and a set of treated with a library of FDA-approved compounds in more than 6 half-log concentrations with 6 replicates per dose for each compound. The samples were then stained with 5 different dyes, HOX, CONE, phalloidin, cyto14, and WGA to detect various subcellular structures. Here you can see the different stain in the GIF at the left side of the slide, and a sample composite image of the data set in the middle. Each of these stains capture a specific cellular structure. HOX targets the nucleus. CONE stains the endoplasmic reticulum. Phalloidin dyes the actin cytoskeleton. Cyto14 tags the nucleoli and cytoplasmic RNA. And last, WGA targets Golgi and plasma membrane. We merged the UV and merged the MOC and the UV inactivated class together since we did not observe any morphological changes in those groups. And then the samples were split into 4 non-overlapping sets: training, validation, untreated, and treated test set. The training set was used for training the neural net, whereas the validation set is used for select and tuning the hyperparameters. The performance of the tuned model is measured on the untreated test set, while the treated test set is solely used for estimating the efficacy of the treatment. Thanks to the special training and inference procedure we have with the MIL, by aggregating and smoothing the patch infection probabilities, we would have an infection map similar to what you see in this slide. Visual inspection of these infection maps, annotated as bright in this figure, suggests that the MIL model is focused on a high intensity stained areas. Further investigation and interpretation of these maps are required to fully understand them, but for now we hypothesize that these are dead or damaged cells resulting from cytopathic effects of SARS-CoV-2 infection. This suggests that the MIL model has incorporated biologically relevant morphological features into these maps, as we expected it to do. Again, notice that in generation of these maps, no form of annotation was used. Instead, the model only uses the sample level labels to localize these infections. Next, in order to estimate the efficacy of a treatment, we first break the treated sample image into patches and fit it into the MIL model. The MIL model provides us with the patch infection probabilities. We aggregate these probabilities across multiple replicates to form an infection probability distribution. Since we do not have any assumption about these distributions, we use the non-parametric statistical test to estimate the median of this distribution and its confidence interval, and then use it as an efficacy score. Essentially, if a treated sample does not have any infected patches, it shows that the treatment was effective. And if the other replicates also exhibit the same results, then the treatment is considered as effective by the model and gets a high efficacy score close to 1. In this slide, you can see the estimated efficacy for sample compound by the MIL model as an example. On the x-axis, we have the log concentrations, and on the y-axis, we have the estimated efficacies. The violin plot shows the estimated distribution for the replicates of the same dose, and the blue curve is a logistic curve fit to the estimations. If the violin is focused at a high value, the treatment is more effective at that dose. Here you can see that the compound was not very effective at the low concentrations, but as the dosage increases, the effectiveness also rises, similar to what you expect to see in an effective dose curve. Well, at this point, it would be totally relevant that you ask why not just use a conventionally trained CNN model for this classification test. In order to answer that, we trained the CNN model based on resident 50 to distinguish between uninfected and infected images. We use the full resolution images as an input, and we use the same data splits as the MIL model to make it a fair comparison. In this slide, you can see the precision recall curve on the untreated test set for 3 models: MIL in green, the conventional CNN in red, and the random classifier in blue. As you can see, both MIL and CNN models are capable of accurately classifying the images. The CNN is even slightly better than the MIL model, indicating that the models have learned morphological features which could be generalized to unseen, untreated test set. However, we need to keep in mind that these 2 models were trained in very 2 different conditions, and their extracted morphological features have drastically different characteristics due to their input and the training procedure. The MIL model has learned to extract highly discriminative features from micropopulation or single cells, where the CNN model focuses on macropopulation of the cells and learn morphological features which are not as detailed as the MILs. In this slide, you can see the same violin plot for 4 well-studied compounds believed to be effective against SARS-CoV-2 for both models. The MIL results are on the left, and the CNN model results are on the right. Although both models have classified roughly the same fraction of the drug-treated test set sample images as uninfected, the results are vastly different. The estimated efficacies by the MIL model are grouped within specific drug treatments, whereas the CNN model estimations are scattered randomly among all treatments and lack any form of structure or pattern. The difference in the distribution of the sample images predicted as uninfected by the MIL model compared to the CNN model shows that there is a drastic shift in the learned feature spaces. We also found that most of the treatments that has been identified to be effective against SARS-CoV-2 by the MIL model is also suggested by other research, by other studies in the research community using different methods, which further suggests the effectiveness of using MIL in this context. It is noteworthy to mention that based on the proposed method, low estimated efficacy does not necessarily indicate treatment ineffectiveness. If the treatment has toxic effects or induces other changes in the cell morphology, the sample may no longer resemble the uninfected or infected class. Subsequently, the model's predictions would not be conclusive, and we cannot control the false negative rate. However, I should mention that if the estimated efficacy is high, then the treatment is effective since it's morphologically similar to the healthy cells. So in summary, in this work, we present a method capable of estimating treatment efficacy of compounds based on morphological analysis. It includes a deep learning model trained within MIL framework to extract morphological features corresponding to SARS-CoV-2 infection in micropopulation or single cells, as well as generating an infection map in a weakly supervised fashion. We compared the performance of the MIL model to conventionally trained CNN model to demonstrate its effectiveness. By integrating a statistical model into the pipeline, the MIL identifies compounds that has been suggested by the community to have antiviral effectiveness against SARS-CoV-2 using other methods, supporting the performance of the proposed method. I would like to thank my colleagues, Kathleen Moriarty, Dr. Olmsted, Dr. Jean, Dr. Nabi, and Professor Liebrecht and Professor Vasson. Thank you for your attention, and I will take your questions now.
Thank you. Very interesting. So there's a few questions in chat. So Li Shen is asking, is the result sensitive to the choice of K, which was, maybe you remember what that was? That's the number of patches, is it?
Yeah, that was the number of patches we used for the training. Well, the performance on the classification task doesn't really change with the choice of K. But if you look at the treatment efficacies, they differ a little bit. And as you increase the K, you see there are more treatments that are passing the threshold that we had for being identified as effective.
Right. And he was also asking, how does MIL compare with attention-based methods?
That's a good question, actually. There are actually some of the attention-based models that incorporated MIL to themselves, so they are not that separated. But in essence, we are thinking that these two are the same concept viewed in two different perspectives. So essentially, they're the same. With the attentions, you can think of having prior knowledge to incorporate it into the model. With the MIL, you can have assumptions on the distributions of the instances and the relation between the samples and the labels that they have. So in essence, they should be like, they are the same.
Okay, thank you. And then Alex Lewis says, great talk. For your efficacy score, how are you controlling for differences in cell density between crops, I guess, patches? E.g., some crops will have more cells, other crops might not have any cells.
Well, that's a good point. We didn't actually control that. What we did was to choose patches that with overlaps, so that we make sure that we got these densities to somehow evened out throughout all of these replicates and the samples. But like that would be a good idea to have some kind of control on that to make sure that we are not getting signals out of the background or any unrelated stuff. Yeah, that's a good idea.
Right. One of the questions I had was sort of related to that, which is, you motivated this initially in terms of, some cells are going to be infected and some won't. And then you went to patches. So have you just really don't want to do cell segmentation, or have you thought about or looked at trying to do cell segmentation, and then instead of patches, you would look at individual cells?
I started to work with the segmentations. The issue that we had at that point was there wasn't a very clear way to do this with the segmentation that really doesn't heavily depend on the stains and the dyes that we have. So we wanted to make it as generic as possible. So we went with the patching direction that can be applied to any other stains or any other combination of dyes.
Yeah. The other thing I was going to ask about was, do you know how long these cells were infected for? I didn't actually know whether they were like they're from patients who have an infection, or whether it was that they're infected in the lab. But if it's the latter, just because you were saying one of the things it looks like the model is picking up is dead cells. So I was wondering if you looked earlier, like when they've only been infected for a little bit, would they be picking up, things that weren't to do with the cells dying, but were to do with the initial effects of the infection?
So from what I got from the Recursion website, and the paper, these cells have been incubated for 96 hours. And we did not get, so they didn't publish any images from the early stages of the infection. So that's not something that we can tell with these images. Gotcha. But I think that would be something technically difficult to do, to have a constant stream of imaging from the whole process of the incubation.
Right. Yeah, and following individual cells would be tough. Definitely.
Yeah.
And the only other question I had was whether you've thought about using, because the other way you might think about doing this sort of thing is to do more of like a generative modeling approach. So you'd have like a generative model for the images that have infected cells versus the images that don't have infected cells. And in principle, you could do something that would almost be like a naive Bayes classification, but using something like a deep generative model for the actual conditional given distribution over images given the infected versus not. I don't know if it's something that people do for MIL. It would intuitively make sense, I think, but I don't know whether
it does make sense. But we didn't went to that approach because our initial guess was that given this infectious map, we would have an easier time to investigate and inspect them visually to see if there is some visual clues that we can get right out of the hand, so we can start working on that. Yeah, but that's also a great idea to incorporate with the MIL.
Interesting thing to try. Great, and we're exactly on time. We have a 20-minute coffee break, so we will see everyone back in 20 minutes. So at I think 10:40 on the West Coast, 1:40 for people like me on the East Coast, and I will let people in Europe will have to figure out themselves what time that is. So see you all in 20 minutes. And thanks to the speakers again.
Sorry, I was on mute. We're not quite co-located at the moment, but mostly New York based.
Right. Gotcha. Okay, it's 1:40 by my watch, so I think we can get started. So next up, we have Tom Serkiu, who's going to talk about neural POTS models. Take it away.
All right, thank you for the intro. So we'll be looking at neural POTS model. And in the field of structural biology, we want to relate protein sequences with their 3D structure. right? So the kind of workhorse approach to do this is, to collect and align evolutionary related sequences into so-called MSA, which we are kind of picturing on the left. So the thing we're going to do then is to fit a model to distill those kind of correlation statistics we're seeing into a pairwise couplings, which define an energy landscape, which we hope can capture the underlying 3D structure, right? So this is kind of like the standard independent POTS model, and it's really like it's a successful method, even the input on state-of-the-art supervised contact prediction methods. So to do this, like this fitting is optimizing a POTS model, right? Like that is how we get those pairwise couplings. So there's a couple of shortcomings and issues with this method, specifically. A first problem is if you have a low depth MSA, right? So if you don't have many sequences in your MSA, you don't think you can have like statistics that are necessary to fit like a good high fidelity POTS model. And that's an important regime actually, because a lot of higher order animals like mammals have very few related sequences in their MSA known to us. And the second problem is that there is no parameter or information sharing between related families. So even if you kind of know that like family 1 and family 2 are related, there's no obvious way in normal POTS model formalism to kind of link that information. And of course, like the second problem is really an opportunity to go and solve the first problem. So we will look at methods to share parameters between protein families. And this idea of sharing parameters between protein families is kind of the core of protein language modeling, which is a field our team has been very excited about. So our method neural POTS model will be building on that work in protein language modeling. So, okay, I'll just do a quick preview, basically an overview of the method. So, okay, we're looking at like an independent POTS model here, right? Like you collect an MSA and optimize a POTS model. Now you do this for one protein family and you do it for a second protein family, etc. So each of those are independent optimization problems. And so what our method and neural POTS model introduces is to basically, kind of skip this whole part at test time. We will just feed in a sequence into the neural POTS model, do a single forward pass, and synthesize or predict those couplings, with a deep model. So basically, we train a model on an intuitive level. We're going to be training a deep model, which of course is going to be a neural network, to summarize, to capture the outcome of all those optimization problems. Okay, so that was the preview. Let us first kind of go a little bit more in depth on the ingredients of a standard POTS model. People also call it pairwise MRF. So this is kind of the formulation of a standard independent POTS model. So we'll look at this equation and at what like goes in it. So this W matrix, like this is going to be the pairwise couplings matrix. So that is a very big matrix of L times K by L times K. So it tells you for each pair of positions in the sequence and each pair of amino acids. It's telling you like how strongly are these positions and amino acids coupled. So this actually defines a Gibbs distribution. So it's an exponential, like log linear model. It is this energy function here is really just a plain like sum over all pairwise terms in W. So it's actually it's a very simple energy function. In practice, we don't want to be computing this partition function like so many places in machine learning where we do everything we can to avoid computing this partition function. In this case, we introduce a pseudo likelihood approximation, which avoids computing, which allows us to avoid computing the partition function. So it's an approximate way. We don't quite fit the likelihood. We fit a pseudo likelihood. It's kind of the current dominant approach. And then in practice, like we're looking here at doing this loss is kind of computed over an MSA. In practice, this finite sample, this MSA is going to be finite sample. So we're not going to have all that many sequences sometimes available to us. So these are kind of some sources of why we may not be able to fit like a perfect POTS model here. Okay, so now we'll give a little bit of background on amortized optimization. That's kind of like the framework how we arrive at this neural POTS model. So it's more general than optimizing a POTS model. It's a really interesting framework that actually kind of is an extension of amortized inference in VAE. But I think it's a very interesting general framework that is giving us a good tool to think about this problem. So where we start with amortized optimization is just a problem, an optimization problem with input X solution. We'll be noting the solution as W star. So we're going to be optimizing, we're going to assume we have several related optimization problems available to us, and that there's some kind of shared structure between those optimization problems and their solutions. So under that assumption, we can think of this optimal solution to define a distribution over X. So as a function over X. Okay, so now we're deep learning people. So what are we gonna do? We're gonna introduce a expressive model to hopefully be able to capture, to learn to approximate this, to approximate this function W star as a function of X. Okay, so this is going to be the objective here. Let me break this down a little bit. So we're going to be able to learn this objective gradient based. So we're going to be able to take gradient steps of this objective with respect to parameters theta. So you can directly compute the gradients here of the main objective. So we'll be optimizing here neural network parameters theta for our optimization objective, like the same as our individual models, but an expectation over sequences. Okay, and we can directly leverage gradients of this loss with respect to theta, the neural network parameters. So same equations, but just with a little bit more of the specifics of the POTS model. And this is really the introduction of neural POTS model. I promise after this, the math will stop. So neural POTS model amortizes the pseudo likelihood loss optimization. So we have this single model, and let me just kind of like talk a little bit through what this equation here exactly means. So we're going to be like just in practice, like a concrete finite sample for a single gradient update step. So basically it comes down to we're going to be sampling some input sequences X. And then for each input sequence X, we're going to be sampling a couple of sequences from its MSA, right? And now we're going to feed our input sequence into neural POTS model to synthesize those couplings, right? And then we're going to evaluate our pseudo likelihood loss on those target sequences from its MSA. Okay, and that's going to be our full loss. And then we're going to backdrop it into the neural POTS model parameters theta, which are shared between all sequences, right? So, and here it's kind of clear how this builds on protein language modeling, where there's also like a single model which takes any sequence as its input. So what do we hope to gain? So this set of ideas that I'm going to be talking about now actually goes beyond normal amortized optimization. So this is specific to our setting where we have a training objective, which is noted here in green, and a generalization objective. So it's a loss that kind of captures the underlying problem we actually care about, right? Like our training objective is basically proxy for our generalization loss, so which would be, for example, how good are we at recovering the structure? So yeah, that's here like a 1D illustration. On the x-axis are just like it's a 1D set of parameters, W. So it's a 1D illustration of our high-dimensional energy landscape. So so the train objective here has an optimal solution, right? We use those hats to indicate it's finite samples, so it's not, it's not optimal. So this optimal solution does not minimize the actual underlying generalization loss, right? Which is always kind of the case or often the case in machine learning. Now our neural estimate, so neural plots model estimate, we reach a different solution than kind of by just directly optimizing the train objective. And now we see that this neural solution is actually doing worse on the primary train objective. This is what we're going to be calling the amortization gap, or it's very related to underfitting. Amortization gap, by the way, comes from the VAE literature. But on the underlying generalization loss, we can actually achieve, we can actually hope to achieve a better generalization despite not achieving the same training objective loss values. So that's what we're going to be calling the inductive generalization gain. So kind of a key to this, which we can't really talk about here, is the inductive bias and the choices that goes into the model. So a different model choice will have different trade-offs, and we'll find different, maybe more or maybe less generalizing solutions. Okay, so let's look at some experiments. And the first set of experiments is really kind of small scale controlled set of experiments. We're using PFAM, this kind of curated database of protein families. We're going to be looking at one clan of PFAM. So in like sequences or families in a PFAM clan are related, but they're not trivially similar. So we'll look at varying the MSA depth, which, on some of the evaluation families, we will artificially reduce the MSA depth. And that's where we will evaluate our neural plots model. And keep in mind, like I'm picturing structures here just to kind of indicate, that they're similar. But during training, there is no structure supervision. So like this method is fully unsupervised, right? It's sequence based only. Okay, so let's look at some results. We have our x-axis being the MSA depth, which I just pictured, and then on the y-axis, we have the precision on contact precision. So looking how good are we at unsupervised structure prediction on the y-axis. And the standard plots model really suffers on reduced MSAs, right? So I just can't figure out, like if you just give, let's say, 10 sequences from the MSA, independent plots model has no way to figure out like what are the right couplings. But neural plots model, at the other hand, is able to do good, like to maintain good performance even on those reduced MSAs. So we're seeing here like it's able to leverage shared structure from related families and from the pre-trained model weights to achieve this inductive generalization gain. And as an additional baseline, just kind of a sanity check baseline, we have this nearest neighbor. So like if we look at like which family in the training set is kind of closest, and like we just use that plots model to predict our structure. So it's a good sanity check that like at some point, like independent plots model does worse than just kind of getting the nearest neighbor in the train set, but neural plots model stays behind across, stays better across the board than this baseline. Okay, so looking at some trajectories over training, this is showing that as we're optimizing, as we're minimizing our primary loss, we see increased precision. So it's a pretty good proxy. And experiments on UNIREF 50. So this is much harder, much more diverse. And actually, let me go to this version. It's the same information. So it's much harder, much more diverse set of sequences, and there's no artificial purging. So on the x-axis, we just create buckets of sequences according to their like MSA depth. So it's the kind of natural, real MSAs. And we see that neural plots model in the low depth buckets also outperforms independent plots models. And here we're building on our high capacity model, which we named ESM1 in the release. So it's continuing training from this very high capacity language model. And then here's some examples. It's nice to look at. In red, like a neural pods model has more hits than like independent pods model, which has more like the misses or crosses. Okay, so let me step back a little bit and think about, we introduced this new objective. Neural pods model learns to kind of synthesize those pods models explicitly as an objective. But it leverages those pre-trained language models under the hood. And more recently, there's been work in our group which is looking at, if we're just interested in contact prediction, structure prediction, rather than kind of synthesizing or like really capturing full pods model energy landscapes. Does that maybe already happen inside of those transformer language models that are trained with mask language modeling? And we were actually seeing like, and we were very surprised that actually happens with very high fidelity. So there was a talk Roshan, our intern, gave at MLCB yesterday, and like he talks about how surprisingly effective, how good these transformer self-attention maps are in modeling and retrieving the context. So it's really interesting that, like, this there's no MSA supervision, there's no structure supervision. You just kind of, purely based on sequences, do this like self-supervised training, and then you look at the attention maps, like the internal self-attention maps inside the transformer, like shake it a little bit, and like these contact maps just fall out of there. This is a really, really interesting result. So yeah, we believe the future is for unsupervised learning, and that's where I'll wrap up. Thank you, and thanks to my fantastic team and like the fantastic interns we've been fortunate to host over the last couple of months. All right, thank you.
Great, thank you, Tom. We have time for a couple of questions. So put your questions in the Q&A. I'll ask a couple while people are thinking. So the W mapping that you're learning, presumably you want to have, you want to be able to handle a situation where you can have different length X, and so you then the W ends up being, well, so if X is length L, right, and then the W you get is KL by KL. Yeah. So are you using an architecture where you have that flexibility to have different length X?
Yeah, that's a very good point. I totally glossed over that. But yes, I mean, these different optimization problems have different, length Ws and different length X inputs. So yes, our model would be, based on on the embeddings of the transformer, which kind of naturally vary with the length of the sequence. So if you put in a longer sequence, you would just have a longer sequence embedding, and then have a longer, KL by KL, L by K by L by K predicted matrix out of it. Yeah.
Gotcha. And since you are seeing that when you have a deep enough MSA, the independent pods outperforms the amortized, which I guess makes sense. Have you thought about, like, in a model-based way, sort of smoothing or interpolating between them? And how you do that? I think actually the same things comes up for amortized VI as well.
Yeah, exactly. Yeah, that's a very good question. So there's definitely interesting ways to go. Like, we haven't actually looked into it too much. So it's just an interesting avenue for future work here, I think. I guess like one difference with amortized, like say this work on like semi-amortized variational inference, for example, would be that so you would expect that kind of amortizing towards closer to the true solution of the optimization problem will always improve. In our case, there's more like a fine balance between sometimes, if you take the semi-amortized idea and you're optimizing your primary objective more, you may actually end up doing worse, like definitely on those small.
Yeah, I think in theory, right? But I remember Dirk Kingma saying at some point that, with the VAE, the inductive bias that's in the Q network actually does does help you, even though if you're being like purely Bayesian, it seems like you wouldn't want that. So yeah, I think it does feel pretty similar.
Yeah,
I guess my last question. Sorry, go ahead.
No, yeah, I agree. Like this is recent work, and like in the VAE setting, I don't have as quite a good intuition. Yeah.
Okay. Well, thank you, and we're just at time, so we will go on to the next speaker, which is Ina Gabasov, talking about split strains, a tool to identify and separate mixed Mycobacterium tuberculosis infections from whole genome sequencing data.
Hi, everyone. I'm going to share my screen.
Great.
All right.
Looks good. You want to go full screen? Yeah. Yeah,
hi, my name is Einar. I will present a tool, Split Strains. It is designed to detect mixed infections such as Mycobacterium tuberculosis and do strain deconvolutions. This work is done jointly with Miguel Moreno Molina, Iñaki Gomez, Maxwell Librecht, and Leonid Chindelevich. So Mycobacterium tuberculosis often occurs as a mixed infection. So for example, we can have a single patient infected by multiple strains of a single organism. However, some of the strains might be resistant to a particular drug, and this might create potential problems for drug prediction analysis. So our goal is to detect mixed infections and deconvolve bacterial genomes. So here we have an outline of a pipeline. We have a mixed infection. We sequence a sample. And then before we start drug resistance prediction, we would like to apply split strains where we learn if the sample is mixed, and we learn proportions and so on. So this comes before the actual drug resistance pipeline. Our goals are identify presence of a mixed infection, estimate proportions of each constituent strain, and we would like to deconvolve strains into separate genomes. So split strains is based on statistical modeling. The whole workflow can be described as follows. So we start with a mixed sample, alignment file, and we identify whether a sample is mixed using likelihood ratio test, and we estimate proportions of each strain using mixture modeling. And next, we would like to classify each read as major or minor strains using naive Bayes classification. And in the end, this will give us two new alignment files, which each representing major and minor strains. So we use likelihood ratio tests to detect mixed infections. So we have two hypotheses: null hypothesis that states that there is a single strain, and alternative hypothesis that states that there are two strains in the data. And likelihood ratio compares two discrete models. So the single strain model looks like a product of binomials. And here we have a product over I, which is a genome site, and D sub I is the number of reads that map to a site I, and K sub I is the number of reads with the most frequent base. And here we assume that there is only one strain, and we allow for sequencing errors, which is just epsilon naught, which is a probability of sequencing errors. And the two strain model is a just slightly more complicated version of a single strain model, where we assume that there is a major and minor strains and some sequencing errors. So the first group of factors can be thought of the probability of a major strain, then this is a probability of a minor strain, and sequencing errors. So we compare these two discrete models, and if the two strain model is the winner, we proceed to proportion estimation. So here is the idea. Use variance size to estimate strain proportions. So here we have an alignment file And we have variance at position I and position J. So if you look at them separately, then we have 75% of A, 25% of T's, and that gives us 75-25 split. And similarly, the side J has a split 87-13. So just by looking at these two splits, we could guess that the major strain proportion is somewhere around 80%. To represent this idea mathematically, we use mixture modeling. So here is the equation that represents the mixture model. We have K strains, we have K densities, and each density representing a population of splits. And we would like to learn a vector theta, which basically describes each of the densities. The frequencies of all variant sites produce two populations. So if we take all the splits that we find in a sample and we pile them together, then we will obtain the following empirical distributions. And using mixture modeling, we can estimate a parametric density of each population. So once we establish the parametric model, we can proceed to deconvolution. So to deconvolve the strains by classifying each read using naive Bayes approach. As before, we start with an alignment file, and we pick a single read, let's say read R, which supports some variants, one variant or more. We keep track of all variants in a special cluster. And we use naive Bayes approach. That is, we compute some number of probabilities of read R belonging to some strain. And we use this data to assign the read to one of the strains. Repeating this procedure for all the reads that support variants will give us two new alignment files. So here are the new two alignment files. So next I'll talk about the results, but before this I'll talk about the data sets which were used for testing. So we have data set A, published by Sobkovac and others. The data set contains 48 mixed M. tuberculosis samples generated in vitro by mixing DNA from two clinical cultures. And the proportions are estimated using spectrophotometry. Then we have data set B, published by Fijao. It contains 16 silico artificially mixed samples with known proportions. And we have data set C, produced for this study. It has 22 in silico samples with 1, 2, and 3 constituent strains. So all these data sets, they have known proportions, known data set B and C have known read labels so that we could check our classification correctness. So the results. Split strain outperforms existing methods in mixed infection detection. So we compare split strains with existing tools such as QuantTB, Mixed Infection Estimator, and MixInfect. So this plot here shows you the receiving operating characteristic. So on x-axis we have false positive detection rate, and on y-axis we have true positive detection rate, and split strain ROC curve, which is a receiver operating characteristic curve, is in green. And as you can see, it is able to obtain a fairly high true positive detection rate, which is associated with fairly low false positive detection rate. So we are doing fairly well. The next closest tool that does also very well is MixInfect. Next, once we detect the mixed infection, we would like to estimate the proportions. And split strains has the higher proportion estimation accuracy than existing methods. So on this chart, we have proportion estimates. The dashed line represents the true proportion, and the dots represent the estimates by all the tools that we compare. And the estimates in green are estimates by split strains. And as you can see, they are tightly clustered around the true proportion. And whereas some other tools, they have quite a lot of spread around the true proportions. And as you can see, when we approach 90 or 95 major strain proportion, then there is some underestimation going on. Next, we would like to look at the deconvolution accuracy. So we measure the separation accuracy by reconstructing the strains' genomes and comparing to their true references. So this plot here shows you two strain samples. That is, on x-axis, we have major strain proportion, and on y-axis, we have error rates, or genome reconstruction error rate. As you can see, when we have major strain proportions somewhere around 90, 80, or 70, the reconstruction error is almost non-existent. That means that the reconstructed strain is almost identical to its true reference genome. However, we start to have a problem when major and minor strain proportions get close to each other. So when we have major proportion 60 and minor 40, then we start to have some errors, 55, even more error. And when we have 50-50 split, then the algorithm fails to reliably separate the genomes. To get more intuition about this problem, we can look at the densities that I showed you before. So when you have, for example, 85% major strain proportion, then we get the following empirical distribution of splits, and our mixture model will look like this. And this is an easy case for read classification. We can easily separate reads and deconvolve the strains. Whereas when we have 50-50 split, then all the splits are overlapping, and the mixture model that we obtain have two densities that have significant overlap. So this creates a problem. So the method cannot, so the reliability of the method degrades as the proportions get close to each other. Split strains can deconvolve more than two strains. So here we have confusion matrices for 3 strains, and the strains are labeled as A, B, and C with respective proportions 10, 25, and 65. We have true label on y-axis and predicted label on x-axis. And as you can see, all reads are classified according to their class or according to their strain. However, we start to have problems when, for example, strains B and C are fairly close to each other. They have proportions 40 and 35, and we start to have some confusion. For example, strain B is confused with strain C, and vice versa. Next, we apply split strain to population level survey data. So we select 59 real samples based on preliminary analysis using a reference mapping pipeline with variant columns against graded databases. In other words, we use expert knowledge, prior knowledge to select 59 samples. And we use this preliminary analysis to detect 27 mixed samples. And split strains detects 24 mixed samples, and all detected samples agree with the preliminary analysis results. So without any prior knowledge or any databases, we can run split strains and detect a subset of mixed infections that were found using expert knowledge. A few words about filtering. So before split strains starts any sort of analysis, we do the following filtering. So first, we would like to work with the genomic intervals listed in the general feature format or GFF file. So for example, if a GFF file has gene 1 and gene 2, then split strains will work only with genomic intervals that are associated with those genes, and everything else is ignored. This is done to avoid problematic alignment regions, and it also helps to maybe zoom in on some regions of interest. Next, if a site depth coverage is below a threshold, then skip it. So as an illustration of this filtering step, if a site has a few reads that align to that position, then we skip this site. This helps to avoid. This also helps to avoid problematic regions in the genome. And in conclusion, split strains is a novel algorithm. It's based on rigorous statistical framework. It can detect multiple strain infections. It can estimate proportions and reconstruct strain sequences. And thank you everyone here at MLCB. And I also would like to thank my co-authors, Miguel Morena Molina, Iñaki Comas, Maxwell Liebrecht, and Leonid Chindelevich, and also NSERC and Genome Canada. And if you want to check out the tool, here is the GitHub link.
Great, thank you. So there's one question in the Q&A that I lost, but it's so it's Mohammed asking, saying, really interesting topic, thanks for the presentation. How can the product of your pipeline be incorporated into a drug resistance prediction pipeline?
Thanks for the question. So I guess one would like to, yeah, I'm just looking for that slide. Yeah, one would like to put it just before the entire drug resistance pipeline. So just right after we get a sample or just a library of reads, we can run an alignment and then apply split strains.
And now we know about multiple instance learning, we can use that as well. I think it would apply. While people are thinking of other questions, I was going to ask a couple. So it feels very reminiscent of the sort of thing people do with whole genome sequencing data from tumor samples, where you're trying to ask if there are, well, first about the tumor versus normal, but then also potentially detecting subclones and the frequency of the or the proportion of the tumor corresponding to different subclones. So would the ideas you have here be applicable there, or other complications?
That's a good question. I'm not sure, because we tried this tool only with tuberculosis samples. And as far as I know, tuberculosis has very specific genome structure, and working with some other cells or organisms might create different results, especially when we do clustering. Then we might not observe something like that. It might be not as clear cut.
Yeah, agreed. Okay, so we've got a couple more questions. So Frankly says, nice talk. Two questions. One, what's your training data set for the naive Bayes? And two, when deconvolving more than 2 strains, does the likelihood computation also change?
The second question is indeed very good. It addresses one of the limitations and future work points. So I'll answer the first question. Naive Bayes is based on this data set that we obtain from looking at each individual read. So having that, we can compute the probabilities, and of course we reuse some of the parameters that were estimated by a mixture model. So that's for naive Bayes. And the second one, the second question is about the likelihood ratio test. So as of now, the likelihood ratio test shows us only, it has only 2 models, a single strain model and 2 strain model. When we have 3 strain models, 3 strain sample, then this likelihood ratio test is not suitable. However, experimentally, we can observe that the output we get for 3 strain models will have a very high likelihood ratio threshold. So that can signify that perhaps there are more than 2 strains. But as of now, we have it as a future work to detect rigorously detect freeze-trained models. There are different methods. For example, one could also look at AIC and BAC when doing Gaussian mixture modeling, but we found that those are often not that reliable, especially when the data set is very messy. BAC and AIC do not give a good criteria for selection of the components.
And Artyom Davidov just wanted to say thank you for the presentation and a very interesting approach. So we're just at time. So I think Sarah is taking over from me on the moderating. Thank you to the speakers. These were all very interesting talks.
Okay, we're gonna move on to the next talk in this session. That's gonna be by hey, I have to find my place here. So that's going to be by Ivan Kofer, and he's going to tell us about accelerated convolutional neural network architecture search for regulatory genomics.
All right, can you see my slides?
Yep.
Awesome, thanks. All right, yeah, thank you everybody for tuning in. My name is Evan Kofer, and I'm a PhD candidate in the Troy and Sky Lab at Princeton University. And it's my pleasure to talk to you today about a collaboration between myself and Frank Zhang, who's a postdoc in our lab at the Simons Foundation. So the title of my talk is just Ambient Accelerated Convolutional Neural Network Architecture Search for Regulatory Genomics. So let's just talk about sort of what that means. So just to give you an overview of the biological problem that we're interested in, which I call the regulatory feature prediction problem. Just in essence, we want to find an accurate mapping from genomic sequences to regulatory features, where a regulatory feature might be something like the binding of a transcription factor or polymerase, histone modification, or the chromatin accessibility at that sequence. The state-of-the-art methods, for this task right now are convolutional neural networks, or CNNs, as I'm sure you've heard many times today. And these are trained, on regulatory sequencing data such as ChIP-seq at a whole genome scale. So beyond just being accurate, though, the flexibility of these models enables a lot of unique sort of potent applications as well, sort of like interpreting, the model with in silico mutagenesis or deep lift. You can identify relatively complicated motifs or even uncover non-additive interactions, between motifs. And then, CNNs have also been used to prioritize non-coding variants, such as SNPs and indels, and hint at regulatory complex components of several diseases. However, effectively identifying the neural network architecture, which is a significant component of all of this that has a huge impact on the performance, is still a challenge. So right now, for most papers, to design their CNN, they follow a really time-consuming manual process that's sort of informally done for the most part. There are a few exceptions out there, but most are sort of manually designed. And this is a visual overview of sort of what that looks like. Sorry. Generally, you just decide on some sort of starting model architecture based on your prior experience, or maybe divine intervention through unknown means, essentially. And this network, we'll call it the child network or the child model for the remainder of the talk. You train this child model, you update its weights, occasionally evaluate its performance on some validation data, and then at some point you record the minimal validation loss associated with the child model architecture. We'll call this minimal loss the reward signal for the remainder of the talk. So after you measure the reward signal for a single child network architecture, you probably will try a bunch of different architectures, again, based on your experience, based on the papers you're reading and whatnot. You'll measure the reward for each of those architectures, and then you'll continue to update sort of your working child model. And then you kind of rinse and repeat this process until you decide on a final architecture. So clearly, this is a very sort of informal and potentially slow and inaccurate process. And I think we can do better and indeed we can do better by leveraging a rigorous optimization procedure. So in neural architecture search, or NAS, we automate the network design process by phrasing it as a machine learning problem. So instead of manually designing the network, we seek to learn which network architectures are best for a given task. So in many ways, this is analogous, to the manual process I showed before, except here we sampled the child network from a generative model, which is generally a second neural network, which we'll call the controller model or the controller network, just to distinguish it from the child model. After sampling several child models, we'll train them all, measure the reward signal for each of them, and then use reinforcement learning to update the controller model based on the reward signal for each child network. And then you sort of repeat this process until you converge. And then finally, just to draw a distinction between the network architecture selection and the actual sort of model production and training, once you've sampled sort of your final child model that you've converged to, you'll then fine tune its weights and probably train it on an expanded training set. This is usually going to be a larger training set than you could feasibly do neural architecture search with. So NAS has proven itself to be extremely powerful in the computer vision domain and produce CNNs with state-of-the-art performance on various computer vision problems. So for instance, here's a plot from a 2019 review article showing the accuracy on the CFAR10 data set for various network architectures. Here the y-axis is accuracy and the x-axis is the architecture in question. And manually designed networks are colored orange, architectures produced by NAS are blue. You can see in general, NAS architectures, outperform manually designed architectures. And just worth noting that this also applies to some other computer vision problems as well, besides what I've just shown here. So recently, the NAS paradigm has been extended to the regulatory feature prediction problem that I mentioned earlier. This was done by my co-author, Frank Zhang. His algorithm, dubbed AMBER, can be used to automatically derive optimal CNN architectures for regulatory feature prediction tasks. And you can read more about this on bioRxiv and try out the algorithm using the code at this GitHub repository that I have in the bottom left corner. So in essence, you provide AMBER a data set for a regulatory feature that you want to be modeling. So maybe ChIP-seq of OCT4. And then it uses reinforcement learning to train a generative recurrent neural network, RNN, controller model that outputs the optimal architecture for the task of predicting OCT4 ChIP-seq from sequence. Here's just a sort of visual overview of the AMBER algorithm. Architectures in this case are represented as a list, essentially of vectors, where each list entry corresponds to a specific configuration setting for a layer in the child network architecture. And the controller model is used to sample different child network architectures, which we measure the reward signal for. And then we, again, use reinforcement learning, in this case, proximal policy optimization, to optimize the controller model based on the reward signal associated with each child model. And then we repeat this process sort of until it converges. However, AMBER and other existing NAS methods are not without their limitations and shortcomings. So first, there are just some general limitations of NAS that we need to consider, and I sort of hinted at earlier when I talked about efficiency in training. So first off, it's really time consuming because essentially to generate one final model, you have to train many child models in search over the network search space. And this can be financially expensive if you're leveraging cloud-based hardware that's sort of exotic, like tensor processing units. And then more recently, people have noted that this can also be very energy intensive and output a lot of CO2 depending on your power provider. So in some sense, it may be unsustainable. And then finally, for the regulatory feature prediction problem, there's this additional limitation because we may want to model different regulatory features in the future. We have to repeat the search process for every regulatory feature we want to model separately. So if you select, say, an architecture for predicting POL2 binding, you'll have to redo things if you want to learn the optimal architecture for predicting OCT4 binding. However, as it seems obvious though that like these two tasks, POL2 and OCT4 binding prediction might have some commonalities, right? But even so, you can't really exploit these commonalities because there's no progress shared across the two separate architecture searches that you perform for POL2 and OCT4, at least with the existing approaches. So if we share progress across these tasks by using a single controller model, we can do better. And this is where our algorithm ambient comes in, which can be thought of sort of as a direct successor and an improvement on AMBER. So here's just a visual overview of the ambient algorithm. And as you can see, instead of performing the search process for a single task at a time, we simultaneously search for architectures for several tasks at once. So instead of just optimizing our controller network to identify the architecture for a single task, we optimize it to produce architectures for any task. So for instance, instead of like in our hypothetical example before, instead of searching for the OCT4 and POL2 architectures separately, the controller model would now sample child models for both tasks. And we would update the controller model using the reward signal from both tasks. And then, to allow the controller model to distinguish between the tasks when we ask it essentially for a child model architecture, we feed it a numerical description of the task as an input when we sample that child model from the controller. So this numerical description is essentially a summarization of the data set associated with a specific task. So it contains high-level information about that task, like the number of peaks in the for instance, ChIP-seq or ATAC-seq data set, or whether a given task is considering modeling a transcription factor or chromatin accessibility or POL2 or histone modifications. Or in the case of transcription factor, the summary includes a little bit of information about some domains, protein domains that may be included in the transcription factor. So as a result of including all of this information, in the controller network's input when we ask it for a child model architecture, the controller network can actually generalize to new regulatory features without conducting a search all over again. So basically, what I'm saying is we train the controller network to generate architectures for a set of training tasks, but we need not update the controller architecture, the controller model to have it generalize and give us architectures for new tasks. We just feed it a description of the new task and it outputs a child model architecture for the task in a constant time. So just to make that like visually clear, here's an overview of the data we use to train and evaluate ambient. So we fit the controller network using 36 training tasks and held out 4 test tasks. For the tasks in the training set, ambient performs a full search over them and updates the controller network weights. But once we move to the tasks in the test set, there's no updating of the controller network at all. We just feed it a description of the task and it outputs an architecture in constant time. So before I move into the results from those test tasks, I just want to try to give you a sense of the policy learned by the controller model by visualizing the architectures that it selects for different training tasks. So as an example, here's a Sankey diagram showing the architectures selected for modeling the EZH2 transcription factor in human astrocytes. To generate the Sankey diagram, we essentially sampled 100 networks from the controller network using the EZH2 dataset description covariates as input. And the vertical stack of gray boxes indicates different configurations of a single layer, so that the height of each gray box is the number of times a given configuration was used for a layer. And the first layer is this stack of gray boxes on the far left, and then the last layer is the stack of gray boxes on the far right. And as you can see, sort of convolutional layers have different configurations, such as the number of filters, the kernel size, dilation, the activation function. And then there's, for dropout, different probability of dropout, et cetera. And just for visual clarity to make this very explicit, here on the right-hand side, I show the architecture configuration that is assigned the maximum probability by the controller model. And as you can see, this in the Sankey diagram corresponds to the thickest band passing through the whole diagram. So here is the Sankey diagram showing the architecture is selected when we repeat this process across all of our training tasks, which is 8 chromatin features 7 histone modifications, 5 POL2 data sets, and then 16 transcription factor ChIP-seq data sets. And this should give you, I think, a clear sense of the policy learned by the controller model. So here, the bands are colored according to the type of regulatory feature modeled in a given task. And as you can see, there are stark differences in which architectures are optimal for different classes of regulatory features. So like TFs seem to have a larger amount of dropout in the earlier layers than the other feature classes. Chromatin accessibility features seem to be best modeled with average pooling rather than maximum pooling in early layers. So really the clear takeaway is that these architectures do vary significantly with the class of the regulatory feature, and the controller model has sort of learned this association. So now I want to talk about Ambient's performance relative to AMBER, which is the existing state of the art for neural architecture search for regulatory feature prediction on the held out test tasks. So here are just the results for YY1 ChIP-seq experiment modeled. This is one of our test tasks, and the x-axis here is the number of hours required to search for the optimal architecture using AMBER, and the y-axis is the reward signal for a given architecture. So the horizontal red line indicates the performance of the ambient selected architecture, and then the vertical dashed green line indicates the time required for AMBER to identify an architecture that performed as well as or better than the ambient architecture. So it's sort of a break-even point. Importantly, the ambient selected architecture is produced in a constant time from the controller network trained on the training tasks. So the AMBER selected one requires several additional hours. And here you can see the results for our other 3 test tasks, where again, it in general requires, greater than 8 hours essentially of searching with AMBER to achieve a child model architecture that was had similar performance to the one that Ambient produced in constant time. So in conclusion here, sort of Ambient is able to consistently generate competitive architectures quickly. So just to wrap things up, some key takeaway points. So first off, Ambient is able to uncover the connection between the numerical task description and the optimal architectures for that task for the regulatory feature prediction problem. And then once we've trained Ambient on several training tasks, it's actually capable of generalizing to new tasks. And for those tasks, Ambient simply takes as input the numerical summary of the new task and outputs the optimal architecture in constant time. And even though Ambient produces these architectures in a very small amount of time, they're competitive with those generated by the state-of-the-art method for neural architecture search for the regulatory feature prediction problem. So finally, I'd just like to thank and recognize my co-author Frank Zhang and my advisor Olga Troyanskaya. This wouldn't have been possible without them. And I'd also like to thank everybody in the Troyanskaya lab for their help and support. Thank you for listening to my talk. I'll take questions now if we have time.
Thank you so much for a really interesting and clear talk, Evan. So there's a number of questions. Let's start from the one in the QA. Okay, so Sanjan Gupta just asked, thanks for the great talk. How is the architecture for the controller network determined?
So the architecture for the controller network in Ambient is actually essentially identical to the one used in AMBER. The only difference is that we have this additional small, densely connected neural network that maps from the dataset summary to the initial input state for the recurrent neural network.
Thanks. And maybe I'll just ask a follow-up question on that. So you showed this plot that for different kind of regulatory genomic tasks, the architectures are somewhat different. I was wondering if you could comment if some of that related to the amount of training data that you have available for the different tasks. So I assume there's much more transcription factor, ChIP-seq data sets compared to some of the other ones that you were showing.
I don't think that, it's possible. I don't, because the tasks are being approached like independently, I don't know how much that would affect it. Um, yeah, because we did hold out transcription factors on a per factor basis. You know, I imagine that there's going to be like enough of a difference between the training and the testing transcription factors that it's not just learning like a general transcription factor model.
Okay, cool. So question from Alex Liu. Do you have an interpretation of why so many of your models seem to prefer max pooling first, then average pooling second? Does this relate to some general property of how features are distributed in sequence?
You know, that's actually something we've been thinking about and trying to figure out since it came up. Honestly, for average pooling versus maximum pooling, the best I can come up with is that maybe when it is using average pooling, it's further sort of exploiting the values of negatives to have things balance out. So it's you know, leveraging somehow like additive signals so that things can sort of zero out, and that's just less important for whatever reason for some factors, and that for those you can just get away with max pooling. But actually, when I also looked sort of at like the computer vision domain of things, I couldn't really get a clear consensus of when average pooling versus max pooling, you know, when which was preferable. So we're sort of struggling to find an analogy there either.
Okay, and maybe just the last one. This is from Surag in the panelist chat window. So how much worse are existing architectures like Bassett compared to ambient models, I guess, determined from similar type of data?
I don't know if it's a fair comparison. We haven't done any direct comparison with this, you know, to Bassett, et cetera, in part because basically our training data for this, each task here is like a feature from the original DeepSea paper. So we probably have to go and make a slightly different version of DeepSea. The real problem is essentially some of our tasks that are in our test set were training set tasks for things like Bassett, et cetera. So that makes it a little tricky to do a fair evaluation. But you know, in the original Amber paper, I believe Frank does a comparison because he's not sort of, you know, sharing, or he doesn't have test tasks in that paper. And I think he does a comparison with DeepSea and Bassett. I can't remember off the top of my head what the results were though.
Okay, thank you so much for a really interesting talk. Now we're going to move to the last talk in this session before the lunch break. That's going to be by Anastasia Belayeva on causal network models of SARS-CoV-2 that explain expression and aging relationships. So please take it away, Anastasia.
Hi everyone, I hope you can see my slides. Okay, great. So my name is Anastasia and I'm a PhD student at MIT with Caroline Mueller. So today I'll talk to you about our work on causal models of SARS-CoV-2 expression for drug repurposing. And actually, since this was a very collaborative effort in our group, I will start the talk, and Louis, who is also very central to this whole effort, will take over the latter part of the talk. Okay, so the goal of our project is to identify FDA-approved compounds for drug repurposing and also enable more interpretability in terms of what these drugs might actually do by providing a way for investigation of potential drug mechanisms and the downstream effects of the drug. So there's been, of course, a lot of prior work in this area. And just to give you kind of a sneak peek of this talk, I would say our approach is a combination as well as an improvement on two major ways of thinking about this problem. So one prior approach for drug repurposing, which has been very popular, is to rank drugs by how correlated they are with the reverse signature of the disease. So we rely on this type of thinking for our computational drug repurposing, but we compute these correlations between the drugs and the infection in the latent space learned by autoencoders. So another popular approach for computational drug repurposing is the analysis of disease networks for identification of drugs. So here you might have a disease network and you might prioritize efforts that are, for example, really central in your network. And we improve upon this type of approach by additionally learning causal instead of just undirected networks. And that this allows you to actually investigate the potential drug mechanism and also prioritize drugs based on their downstream effects. So in our work on SARS-CoV-2, we have specifically tried to address the age-dependent SARS-CoV-2 pathogenicity. So as many of you know, there's a huge age-dependent difference in the effect of COVID-19 on individuals, both in terms of hospitalization rates as well as the mortality rate. So the focus of our work was to specifically identify drugs that target both of these pathways. So how did we do this? Well, first we wanted to see what are the genes at the intersection of SARS-CoV-2 and aging pathways. So we obtained this bulk gene expression data of A549 cells, which are lung cancer cells, and also these A549 cells infected with SARS-CoV-2. So from this, we can obtain differentially expressed genes from that data. And next, we also looked at gene expression data of the lung in young and old individuals based on GTEx. And we were also able to identify differentially expressed genes associated with aging. So next, we intersected these two sets of genes, and the intersection was statistically significant. And moreover, when we looked at the direction of the log-fold2 change in this intersection, we saw that a lot of the time the change in gene expression had a concordant direction. So, for example, like either both genes go up together or they both go down together, but there is not much mismatch in terms of the direction of the log-fold2 change. So that additionally suggested to us that maybe it's important to look at SARS-CoV-2 and aging together. So now let's talk about how we actually approach the task of drug repurposing. So as I mentioned, one really popular approach in this area is to find drugs that are maximally aligned with the reverse of the SARS-CoV-2 infection. So the first thing that we did is we wanted to see if we could identify the signature of SARS-CoV-2 robustly. So as I mentioned, we had all of this different data of A549 cells, as well as A549 cells with ACE2 receptor, different batches, different rates of infection. And across all of these different experiments, this is shown in this PCA plot, we were able to robustly identify the signature of SARS-CoV-2. So now let's talk about where we actually get the drugs from. Well, there's this really nice data set from the Broad, and it's publicly available, called the CMAP. And this data set contains gene expression profiles on around 1.2 million samples, and they're treated with thousands of different perturbations, including about 800 FDA-approved drugs. And it was also measured on many different cell types, and actually including this A549 cell type, which we have the SARS-CoV-2 data on. So one simple approach to drug repurposing would be to take the A549 cells from the CMAP data and look at all of the drugs that we applied to, that were applied to A549 cells, and then just pick the drug that aligns best with the reverse signature of SARS-CoV-2. So of course, the simple approach is throwing away a lot of data because there's cell types, there's some missing data in the CMAP data set, so not necessarily all of the drugs were applied to all of the cell types. So by just considering A549 cells, we might be throwing away some data. And perhaps more importantly is that we don't actually specifically care about A549 cells, but we want this whole analysis to be transportable to, say, AT2 cells or other kinds of cells that are in the body or the lungs that are actually affected by SARS-CoV-2. So now the question is, how do we predict the effect of a drug on the cell type where this drug effect has not been measured? So how do we do this? Well, autoencoders is what we're going to propose to use. So a lot of you have probably seen a lot of work on latent space arithmetic or style transfer using autoencoders. And it's also a similar idea here. So just as an example, suppose we have a data set of people with smiling faces and also neutral faces. So I can embed these images in the latent space using an autoencoder. And in this latent space, we can learn a vector that corresponds to making a person smile. So now if we take another person, and if we only have an image of them with a neutral face, we can embed this image into the latent space, apply this vector that corresponds to making a person smile to this point, obtain another point in the latent space and decode it back to get an image. And you'll see that it works out nicely. This person is actually smiling. So similar ideas have been applied in the world of single-cell RNA-seq, for example, as well, for predicting the effects of perturbations. So this idea suggests that maybe we can do something similar. So I might have normal cells for cell type 1 and treated cells, cells treated with drug A for the same cell type. But for another cell type 2, I might only have data for these normal cells, but not these cells treated with drug A. So to actually obtain treated cell type 2 with drug A, we can just transport this, apply this learned effect of drug A on this other cell type. So of course, this only works if these drug signatures are actually aligned across cell types. And so we wanted to test whether with our framework, that's actually the case. So now remember from CMAP data, we actually do have a lot of data on drugs that were applied both on cell type 1 and cell type 2. So I can actually look at the correlation of these drug effects in the latent space across cell types. So the plots here show the correlation between drug effects of A549 cells treated with drug I and MCF7 cells treated with drug I in the latent as opposed to the original space. So each point here corresponds on these scatter plots corresponds to a different drug that is both available for A549 and MCF7 cell types. So you can see that with an autoencoder, and specifically an autoencoder that has higher as opposed to low embedding space, we obtain an alignment of drug signatures across cell types. And as opposed to PCA, which of course throws away a lot of signal from the data, you also get a good reconstruction of the gene expression data. So we performed similar analysis for all other pairs of cell types in the CMAP data and observed similar trends, which is shown here.
Okay, so I'll take over from here. Can you hear me, Anastasia?
Yes, you're good.
Okay, so basically after embedding the perturbation data using the autoencoder, we compared, as you can see on this figure, the drug signatures in the latent space with the reverse signature of SARS-CoV-2 infection. So here you can see this green arrow that points from infected cells to non-infected cells. that denote the reverse signature of SARS-CoV-2 infection. And we basically want to find those drugs that have a signature which is maximally aligned with the reverse signature of SARS-CoV-2. And by doing this, we identify about 140 FDA-approved drugs that have a high correlation with the reverse signature of the disease. And we show a sample of them on the right table on this figure. And one interesting thing is that among these drugs, some of them are already under clinical trial right now, such as ribavirin or quinapril, for example, and others. Next slide, please. Can I have the next slide, please, Anastasia? Cool. So now that we have these 142 drugs that were selected and they're potential candidates for drug repurposing for SARS-CoV-2 infection in these A549 ACE2 cells, we would like to filter these drugs down even more to select first the drugs that have an impact on the combined SARS-CoV-2 and aging pathways, and also to select drugs that we think are going to be effective. So this is what we're going to do now. We'll start first from the 219 genes that Anastasia mentioned that lie in this intersection between the SARS-CoV-2 infection differentially expressed genes and the aging differentially expressed genes. And for these 219 genes, we will only select those that have concurrent gene expression in the two pathways, meaning either upregulation in both or downregulation in both. So we end up with about 160 genes. And we will look at how these genes are connected within the general human protein-protein interaction network. And we're going to do more than this. We're going to actually try to find a minimal subnetwork in the human protein-protein interaction network that connects all of these genes. And we do this using a technique called price collecting Steiner trees, which allows us to find a reduced interactome of about 250 genes that include all of these 160 differentially expressed genes and other genes that connect them in the network. So now that we have this network, which you can see on the lower left of the slide, the resulting, we will intersect this interactome, the gene in this interactome, with the targets of the candidate drugs that we obtain from the autoencoder analysis. And we will only consider here the drugs if they have a high affinity for their target as measured by some affinity constant. What is interesting here is that the resulting drugs, which are shown on the right, and it's a bit small, so I'm just going to describe quickly, is that most of these drugs are actually protein kinase inhibitors. So it's a class of drug that usually has the suffix tinib, so dasatinib, axitinib, for example. And they target mostly serine threonine protein kinases and receptor tyrosine kinases. Next slide, please, Anastasia. So we have, kind of filtered down a bit our list of drugs, but now we're left with a problem, which is that, a selected drug for us is important and irrelevant only if it has a downstream effect on the genes that are differentially expressed due to SARS-CoV-2 and aging. So if it does not have an effect on these genes, then it's probably not going to be very effective. The problem is that the protein-protein interactome that we have is actually undirected. So that's the upper figure that Anastasia is circling right now. Basically, the drug target here that we're considering is the green diamond. It is connected to a bunch of differentially expressed genes, but we have no idea what is the directionality of these edges. So if we are in the case that it's the center, like the middle picture here, where all differentially expressed genes point to the green diamond, which is the drug target, well, if you intervene on this target, you will not change probably the expression of the differentially expressed genes. So a more desirable situation here would actually be the lower picture, where the drug target is upstream of most, if not all, of the differentially expressed genes. So intervening on this drug target will actually have an effect on the differentially expressed genes. So to orient at least partially some of the edges in the protein-protein interactome that we have, what we're going to do is we're going to use causal analysis. And in particular, a technique that was an algorithm that was developed in our group, greedy sparse permutation, or GSP. And this algorithm allows us to orient some of the edges. And from that, we obtain that we have several drug targets in our pool that have downstream effects on differentially expressed genes due to SARS-CoV-2 and aging. And the most important drug target seems to be RIPK1, which is the largest number of downstream genes. And note that this was done, like this causal analysis was done using single cell RNA-seq on A549 ACE2 cells. But we also repeated this with AT2 cells, which are like an actual cell type, epithelial cell type of the lung, which has been shown to interact with SARS-CoV-2, and we obtained similar results. So like in the end, what this means is that SARS-CoV-2 is probably a key target, against SARS-CoV-2 infection with a highly age-dependent role. And that's interesting because it has been shown to bind with SARS-CoV-2 by a recent study. And it has also been found to be a member of an age-dependent module. Next slide, please. So that brings us to our conclusion. So in this work, we developed a novel drug repurposing platform that integrates several data modalities. The first one is transcriptomic. We also use protein-protein interaction data. We also use drug target data. And one thing that has been instrumental in our work was perturbation data, like the CMAP dataset. The main techniques that are used here are autoencoders to basically align drug signatures in the latent space across cell types, and Steiner trees and causal analysis to filter down the drugs that we want to consider. So I'd like to mention here that this platform is computational, as you can see, so it would require further biological or clinical validation, which we have not done in our work. But in general, I think the main message is that this platform was applied to SARS-CoV-2 here, but is broadly applicable beyond this SARS-CoV-2 example. All the data sets we used are public, and all the steps are detailed in our paper. And with that, I'm going to thank you for listening, and we're happy to take any questions.
Thank you so much to both of you for an interesting talk. And sorry, I didn't mention initially that there'll be two presenters for this talk.
That's fine.
Okay, so let's get to the questions. First is from Li Shen. How can you make sure the embedding is biologically relevant, since, I mean, all the model knows is about reconstruction loss?
We try to, as I mentioned, like we try to see whether, for example, drug effects are aligned in terms of cell types. So that's one way we try to see that like what you learn is actually relevant.
Okay. Yeah, I guess that's a complicated problem to address, but we always need proxies to figure out if what we're seeing are biologically meaningful. So Li Shen also has a second question. Can you select multiple drugs that jointly maximize the reverse effect?
Yeah, I don't think we did this. We did not do this in this work, but there may be, maybe Anastasia, you know ways to do this, but we didn't do it in this work.
Okay. Anonymous attendee, why did you focus on aging pathways?
Well, I mean, it's just from like data on COVID-19, like people have shown that obviously it affects older individuals much more. So, and there's been like a number of hypotheses in terms of like why that's actually the case. And some of the hypotheses involve like this idea that there is crosstalk between SARS-CoV-2 and aging, either like because of immune system, or there's been other hypotheses regarding like mechanoregulation of cells as they age. So that's why we thought it's important to actually look at both.
So actually I mean, if I understand correctly, it's not, you're looking at genes that are differentially expressed by age, right? That's
yes.
correct, is that right? Okay,
yes.
so it's not, I guess, per se aging pathway, but genes that have some sort of differential expression as a function of it.
Exactly, yes.
Okay, so last question. Why overlap aging and SARS-CoV-2 differentially expressed genes instead of just SARS-CoV-2 differentially expressed genes?
I guess it's similar type of answer. Just because people have shown that there is an interaction between SARS-CoV-2 and aging. So we thought it's important to actually model that. And also, like, maybe more broadly, it would be more important to have like some kind of analysis or some kind of drug repurposing for people who are like more affected by this.
Right. So in a way, you're using like biological, I guess, hypothesis to reduce the search space and the number of genes that you're going to investigate.
Yeah, I think that's one way to think about it.
Okay, so last question, Jeremy Zucker. What did you use to orient the edges when applying GSP? And second, did you validate the edge directions against known sign-directed protein-protein interaction networks such as OmniPath?
So here, yeah, this is a really good question. So in general, like causal structure discovery methods, they work by essentially inferring from data conditional independence relations between genes, and you can use those to assign some edge orientations. So here we didn't validate this method against the specific directed PPI that was mentioned. But what we did do is the CMAP data, it's actually super nice because in addition to drugs, it actually has additional perturbation experiments. So there's like data on overexpression of genes and knockout of genes. So if you think about a causal network, well, what it predicts is you can use it to predict an effect of an intervention. So if you like knock out a gene, you see what are the downstream effects, and these perturbation experiments from CMAP data, they are essentially the ground truth of what are the actual downstream effects when you knock out or overexpress a certain gene. So we actually use this to validate our method on A549 cells using these perturbation measurements. And you can see there's a, if you look at our paper, there's a plot about that.
Okay, thank you so much to you both and all the presenters in this session. Before we break for lunch, James has an announcement to make about the exciting panel that's coming up.
Hi, everyone. Yes, so we have a panel discussion in 23 minutes on machine learning for molecules, proteins, and drug discovery. I think it will be quite a fun and interactive panel. So we encourage people to, if you have questions or topics that you would like to hear us discuss, please enter that into the Q&A function during the break. You're also welcome to enter them in as the panel progresses in real time. But if you enter it before the break, we can be sure to prioritize those questions. So thank you, and look forward to seeing you in a few minutes.
James, one thing we should do, there was one student who missed out on the Spotlight presentation yesterday because she was not able to sign in for some reason. So I thought we would just give her 3 minutes before the panel discussion just to present her Spotlight.
Okay, would it be okay if we do that after the panel?
Sure, we could do that too. I don't see her signed in right now, but she was signed in the morning. So let's see if she shows up.
Okay, sounds good. Hey, Rich, Lucy, Mohammed, thanks for joining. We'll just give people another couple minutes to come back from after lunch. Okay, great. We can get started. Hi, everyone. Good afternoon. Welcome to the panel discussion. So the goal of this panel is to discuss machine learning applications in molecules, proteins, and drug discovery. I am James Zoe. I am assistant professor at Stanford, and I'm the moderator for the panel. So we're super excited to have three leading researchers, experts joining us for the panel discussion. We have Rich Bono, Lucy Colwell, Ashi. So maybe we'll start by just asking each of them to introduce themselves, maybe say a little bit about your backgrounds, and also a little bit about what is your research program in your group. Let's start with Rich. Do you want to go first?
Sure. Thanks, James, and thanks to the organizers. So my background is in protein folding, and also we do a lot of work with regulatory network inference. I think for the purposes of this panel, the work that we do that's most relevant is work to integrate protein structure and function. And so that's work that combines graph convolutions, self-supervised methods for extracting features from proteins, and other methods. But the structure function axis, I think, is one of the things I'm most excited about and excited about talking about today. So.
Great, thank you. Lucy?
Sure. Hi, everybody. My name is Lucy Colwell. I'm a faculty member in chemistry at the University of Cambridge. I'm currently actually on secondment at Google Research, where I'm lucky enough to get to work with a bunch of people who are sort of more machine learning experts. And my background was originally in math, and then worked on a bunch of problems, kind of the intersection of like applied math and bioinformatics. I did some work on structure prediction a while back. That was kind of fun. But currently, I'm really interested in the problem of being able to go from sequence to function. So being able to sort of potentially jump over the structure prediction part and go straight from sequence to function as measured in experimental assays. And we've also done a bunch of work on like protein ligand binding or protein interactions, and sort of like putting this all together, I think is a current challenge, given the huge amounts of data that are available and the exciting models that people are developing, a bunch of which we've heard about over the last couple of days. So yeah, thanks to the organizers. I'm kind of interested to see what topics we cover in the next few minutes. Thanks.
Great, thanks, Lucy. Mohammed?
Yeah, thanks again for the invitation to speak. It's a pleasure. So I'm an assistant professor at Columbia in the systems biology department. I guess my background is sort of really straddled computer science and biology throughout my training. Most recently, I was at the systems biology department at Harvard, and then I moved here. My lab's focus is probably sort of twofold. One is developing methods, or really kind of method development, machine learning method development for molecular systems, both protein structure, protein function prediction, but also protein-protein interactions, protein-ligand interactions. So trying to sort of build networks in a bottom-up way from using machine learning methods. And then the second sort of part is really about using these types of molecular models to sort of probe questions in systems biology. Both on the sort of basic level, so things having to do with signal transduction networks, how they're organized sort of across metazoan scales, as well as kind of more translational questions of trying to use these molecular models to sort of relate genotype to phenotype, to kind of translate information from, coming from various kind of modalities and whole genome sequencing, that sort of thing, into sort of modalities that are instantiated at the level of, say, networks, and using those types of networks to inform, for example, patient drug response or prognosis prediction.
Great, thank you. So we'll start with a few questions, and I also encourage the audience to, if you have any questions, please type that into the Q&A box, and then we'll monitor that and then direct that to the panelists. So the first question is maybe just sort of an invitation for each of you to perhaps just say a little bit more about which particular research direction, under this broader umbrella of topics, are you currently the most excited about? All right, maybe we can also start with Rich again.
Oh, thanks. Well, right now, I'm very excited by a lot of the methods that we've been using to predict things that are crossing over into the generative. And so I'm really, and I think based on what I'm seeing on bioRxiv, I'm not alone there, but one model that we're really excited about, and it's sort of a class of models, are models that, with a functional constraint from a model that learns how to predict function, models that can generate new sequences. And I think, we've seen a lot of really cool work that can generate sequences that fold to a desired structure. And I think soon we'll learn how to actually become a little bit more multimodal, a little bit more in control of these sort of new feature extraction methods. And we'll be able to actually get something that has a fold, but also has a function. And I don't think any of us are really there in the general sense where you can just pick 3 things out of the geo ontology and get them on a fold of your choice, where and probably that's not possible. But there's some really exciting stuff, I think, going on in generating new sequences, and I hope to hang on for dear life and be part of that research, because it's moving. It's a fast-moving field, and there's just so much amazing work going on there. So I think that's the thing I'm most excited about right now. But I think I'm also very excited about just the prospects for protein function prediction with a lot of different methods. There's not just protein methods, but there's also methods to take lots of other considerations like networks and things. And I think there's actually going to be a lot of discovery of new enzymes and new functions in metagenomics data sets and things like that from being able to annotate without alignment, essentially. And so that's another thing that I think we're very excited about. That might cross over into the not relevant for this panel, but yeah, I think those are my 2 favorite things right now. And I guess not just for me, but also in terms of what I see popping up on bioRxiv and all these things as well. So
that's great. Yes, we'll definitely want to come back and talk a bit more about these generative models and the promise and challenges there. Yeah, Lucy.
Yeah, actually, to pick up on what Rich was saying, I'm quite curious about these generative models and sort of partly the extent to which we really kind of need them, and the extent to which they, at least in their current form, produce good sequences. Like, I think that the sort of vision of being able to use a generative model to design totally new sequences is really exciting. What I see so far, hasn't sort of solved that problem. So I'm very excited about it. I also think that discriminative models, or models where you score a bunch of potential sequences and use that way of choosing them, are very powerful. And so I'm sort of excited about, translating those sequences into wet lab experiments and, figuring out how we can optimize those models, without necessarily needing them to be generative. It's sort of boring, but I'm keen to see what happens and how that can help us walk through sequence space. I guess, in terms of being able to design totally new proteins, that's really exciting. Like, if we could design enzymes to catalyze reactions that so far can only be catalyzed by heavy metals, for example, that would be really useful. That seems, the idea of designing new folds or being able to sort of select folds and sort of sellotape function onto them, that would be really cool. It seems very much in the future. And being able to sort of really redesign interactions between proteins seems very exciting and kind of eminently plausible, honestly. Like, if we're just, we sort of are just thinking about the binding surface, the interactions between proteins, I think there's a lot of traction there, but we're not really necessarily modifying the fold or the function, but we're simply designing things to stick together. That seems kind of tractable right now, maybe easier. But yeah, overall, exploiting this whole sort of design, make, test loop where we can really synthesize exact sequences, test them in the lab rapidly, design new assays and get information, feed it into models. Like, I'm really excited about that side of things in particular. Yeah.
Very interesting. Great. Yes, Mohammed.
Sure. Yeah. So I think I definitely think generative stuff is amazing, and there's been a lot of really cool work in that space, which I think will continue to be the case. But I'll probably take a slightly different focus from my side, and what I think really excited about is just the idea of using structure and as a way to sort of probe questions on a systems-wide level, right? So the question I'd like to ask people often is like, imagine protein structure prediction is solved tomorrow, right? So we can predict the structure of any protein we're interested in, and structural information becomes as abundant as sequence information. Well, how would that change what we do? In terms of the analysis that we perform, and particularly from my side, things like single transaction networks. Right? If we can understand, how these complexes form on a structural level, does that resolution sort of allow us to probe these questions at a level that we haven't been able to do before, and maybe to kind of begin to uncover the logic behind how information processing is done in the cell at a molecular level by understanding these things as molecular machines. I think on a practical level, what that means, for one thing, as the corollary of this, is that I think methods that have to do with structure representation learning suddenly, I think, will become very important, right? If structure is sort of widely available and abundant, then suddenly the availability to sort of input and reason and process structure becomes really key. And so that to me just seems like a really interesting area where a lot of it could still be done. I think there's been a lot of emphasis on sequence learning, on graph learning, but really sort of applying methods sort of essentially on geometric objects, things that exist in 3D space, would be sort of a very, very kind of natural next step. And I mean, moving a little bit further afield, I don't think we're quite there yet, but certainly one can imagine in a few years that maybe even protein-protein interactions are solved. Right? So we can predict an interaction for any given two proteins. And so if that's possible, what can we do? What sort of systems biology can we build based on sort of abundant molecular information? So I don't think we've sort of, certainly I haven't kind of grappled with this question myself, but I just feel like it's something that will really alter their paradigm moving forward.
Great, very interesting. So I think that there are two very interesting threads that everyone mentioned. One is related to these generative models, another is related to maybe incorporating network information and systems biology to learn more about these functions. So certainly love to discuss both of these threads. So picking up a little bit more on the generative model side, I think there's a lot of interest, especially among this conference participants in that topic. So I think people will be quite interested to hear your thoughts on where are we in terms of the current limitations of using generative models for new, let's say new drugs, new molecules, new proteins. So how close to reality are we to actually really design things that have this feedback loop where we can synthesize them and deploy these? If anyone wants to jump in, please feel free.
Yeah, I guess the synthesis challenge for small molecules is pretty significant, and we're seeing really exciting work, around trying to sort of address that, in current conferences and in the literature. But I think it's a significant challenge to be able to, really figure out how to make the molecule that your generative model suggests, right? And in fact, whether you can make it or not. I think that is still a pretty significant challenge. I don't know how much people here are familiar with that literature, the sort of models for chemistry, models for molecules. I think both sort of the path, sort of the reactions involved in making those molecules, and also the conditions that you would need in order to get a reasonable yield. Like, that's a pretty unsolved area.
Yeah.
And I think there's a lot of potential. Actually, if people are interested in that area, there's a lot of work to be done. And it's there's people working in the area, but it's not that crowded. So there's an advert here for we should consider chemistry. It's cool.
Yeah, and I think that actually leads into kind of one of the things that I wanted to point out is if we're talking about drugs, changing things, if we're talking about either drug design, small molecules that change proteins, or maybe small domain mimics, or even little fragments of antibodies, or if we're talking about enzymes, proteins changing small molecules. Either way, one of the things that we've run into, and when I say we, I forget it. It's everything I've read lately, but I feel as if there are a lot of people who are making really nice advancements to predict and function and structure and unite that. But then the small molecule space, we have so few examples of each small molecule interacting with each protein that there is going to be this. Well, I'd be interested in what Lucy and Mohammed have to say about this idea that the chemical diversity, when it runs into proteins, will have probably great ways of growing out graphs and deciding if they're synthesizable, maybe for chemicals. And so we might get a control of that, and then also have some nice ways of dealing with protein structure function, but we still might have a further challenge bringing them together because the interactions involve the orientation of the molecule and many things that maybe I haven't yet seen or haven't thought through. And so I sort of feel like one challenge is that we have a good understanding of, or we could have a good understanding of these vast databases of small molecules and or at least begin to get at what Lucy was talking about. And then proteins, Mohammed can fold proteins, we're good there. But then if we try to put these babies together, it's, we still have something where we don't have as many observations. We don't have as much, well-controlled data. But I that's something I'd really want to hear, the other two weigh in on as well, because I think I'm just saying that it's scaring me. I'm not really offering a lot of, direction there.
I'll echo this just a little bit. I mean, I think you're right, and I think fundamentally the problem is that chemical space is much bigger, right, than protein space. So in some fundamental ways, it's much more constrained. And one other point I would add is much of the data we have comes from things that we synthesize and medicinal chemists have synthesized, which look very different than natural products, for example. And if you really think of kind of the broad spectrum of chemical space, there's just so much stuff that we essentially haven't even begun to sample. So that there are real kind of serious distributional sort of biases in what we have that I think are going to be a challenge. But on the promise side, I guess, one thing that I've been sort of quite keen about is seeing just incredible level of development on the quantum chemistry side of things, right? Speeding up things like DFT and coupled cluster theory and all that stuff to scales where they've almost become like classical, force field type model. And not quite, we still call that an order of magnitude away, but we're getting there. And so that, and I think it's good, broadly speaking, in terms of to combine deep learning, reinforcement learning, all these techniques with really sort of essentially simulators or in silico generated data coming from sort of these high level theories. That just seems like a very sort of inexhaustible well of potential, right? Because it's essentially taking compute and turning it into data, right? And maybe we're not quite there yet, and maybe the scaling isn't quite there yet, but it just seems like it's a direction where over the next 5 to 10 years, we'll get to a point where we can potentially get information that we just are experimentally very difficult to observe.
So you're sort of suggesting that these models where you can very carefully choose what you simulate. You do all this computation to essentially then populate the parts of it of the maybe the space of proteins and molecules interacting, and then from there, yeah. So you're saying that more expensive classical or quantum methods, even if they're ML enabled, you'd use those to sort of build up some of that experience, even if it's not in an already existing data. So that's sort of what you're suggesting for that.
And I mean, there are things that you just can't probe experimentally too. You know, you can't measure the energy of two particles almost on top of one another because it's just too high, right? So there are things there are parts of the potential, whatever you're trying to learn, that really can only be probed computationally. And if these methods, you know, scale like into the 7 or something, so they're horrible, right? But if we can use machine learned versions of them that scale like n squared, then we could pretend, and they are very accurate. They're comparable to experimental data, then we could be very judicious in generating a handful of data points that really fill in the missing pieces. I mean, handful. it's going to be long, but it's essentially a kind of a much more active learning type of approach that's intelligent about generating data from a expensive computational process.
Yeah, that's. And I know in physics there's been a lot of success with that. So I mean, that is a really interesting approach.
So do you think the molecular, I guess, simulations are accurate and efficient enough now to use the simulators themselves to train the machine learning models?
Not yet, no. I mean, we still have a long way to go. But in terms of, say, computing the energy state of a fixed geometry, the quantum methods are pretty accurate. Not running a dynamical thing, but just taking a single snapshot, right? So, but these are extremely expensive. I mean, the raw methods, they don't scale beyond like 50 electrons or something. So it's very, very, But the point is there's been tremendous progress in making them scale and get faster and faster by using machine learned, essentially, kind of proxies for the underlying theory. And so it seems to me like, you know, the issues that Rich was bringing up, they're fairly fundamental, and I don't think actually data will save us. It's that we just can't generate enough data because chemical space is so large. So on the long-term horizon, I feel like these methods, once they're made to work, they're not there yet, are our sort of best long-term kind of chance to really solve this problem.
I guess I'm super interested in how we can really use experimental data to kind of calibrate and validate these methods. Like I think, you know, as Mohammed says, there's a lot of it. It's been a huge amount of exciting progress, and somehow being able to constrain these methods, calibrate them with experimental measurements, like I guess something kind of has to happen, right? Because we need to know that we're not just living in sort of, you know, some kind of artificial universe. And so I'm really excited to see more work on that end of the spectrum, in order to sort of calibrate the simulations and really make sure we can understand them. In terms of, you know, capturing chemical space, like it is huge. And I think, you know, one approach is to sort of try and sort of fill in the gaps, and another approach is to really sort of specify the limitations of our models and to understand them. And, you know, these things obviously go hand in hand, right? Like, you know, we sort of have some data and we know that we can build models close to that data, maybe. You know, I sort of always come back to the idea that even interpolation in chemical space is difficult, right? Because like it's just such a kind of crazy landscape. It's not, things are not smooth by any means. And so trying to figure out, you know, where we can trust models and how we can get a better idea using like attribution approaches or using uncertainty. I think there's a lot of work to be done in that realm, trying to figure out really where we can trust models and then experimentally validating that. Like it's so easy in this sort of chemistry area to convince yourself that you have a great model that predicts accurately on your data splits, and then to realize, you know, afterwards that there was an issue with your data split, or, you know, it wasn't temporal, or I don't know, there's like, yeah, even just that your model works really well, you know, for some data, but as soon as you actually test it in the lab, for whatever reason, it doesn't hold up. And I think getting standardized data is really important in this respect, and the field has a lot of work to do there. Like in bioinformatics and in protein structure in particular, things are beautiful, right? We have the PDV, it's amazing. We have all these sequence databases, it's great. And in chemistry, it's as I think Mohammed or Rich said, you know, molecules are picked by medicinal chemists to be tested. Like it's very non-standard, and protocols vary between labs, and everything is a little bit less well organized. So being able to trust data sets in the literature is kind of tricky. And my you know, chemist colleagues sort of shudder at the idea of like collecting all this data and like integrating it and building models. So to them, that's like an anathema. So that's also a big challenge.
That's a very good point. I mean, I think one issue that's really almost somewhat unique to chemical data as opposed to say protein data even is that it's actually just hard to to do splits, right? Because it's, I mean, the notion of chemical space and distance in chemical space is a little tricky. And most of the data is coming from pharmaceutical discovery programs where you have a congeneric series and there's all these, scaffolds that are very similar and there's just a few things that are different. So it's extremely biased in sort of very, recalcitrant ways. So I feel like basic questions about how do you actually just do your training are still pretty outstanding in this space. I agree. Yeah.
Yeah. Now we have a question in the Q&A, James, that might be kind of related, which is how does evolution help us predict protein function and structure? And I guess I could start by saying that it can also hurt you in the grand scheme of things because it makes it very difficult. I think Mohammed just mentioned that when you look at the distance between different chemical structures, you have to use a sophisticated distance metric. You have to think about it a little bit. And when you look at the distance between two proteins or two genes, you have to really think very hard to make sure that you're not looking at the same thing. So I guess I would, just let a question float, which is, if you don't recognize the homology between two things, and one of them's in your training set and one of them's in your test set, does it still count as accidentally overestimating your performance? Because in some degree, if your model can pick up on that homology, then aren't you doing something that BLAST can't